Shall we start, sir?
Yes, madam.
Okay.
Yes, please start.
Good morning and a warm welcome, Honorable Principal of our college. Welcome, Dr. Richa Ashma, Madam, Associate Professor at Department of Zoology, Savitribai Phule Pune University, and participants for the first session of second day of this faculty development program. It is a privilege for me to introduce today's guest, an alumnus of the Jadavpur University, Kolkata. Dr. Richa Ashma received her doctorate in science from Central Forensic Science Laboratory, Kolkata in 2007. Let me share you. CFSL Kolkata is the first DNA profiling unit in India and is credited for analyzing several important national crime cases. The lab was declared the Center of Excellence in Biological Sciences in 1998 with a state-of-art forensic examinations and research facilities. As a Directorate of Forensic Science Research Fellow, Dr. Ashma worked in the third area of forensic molecular biology, wherein she used STRs or microsatellite markers as a tool to profile seven Indian communities. While performing the study, she got fascinated to know whether microsatellites, which are mostly present in the non-coding region of the genome and considered very useful for DNA fingerprinting and human identification, are just genomic cushion or whether they also help an individual in combating disease stress and have cause-effect relationship with disease predisposition. Currently, she is an associate professor in the Department of Zoology, Savitribai Phule Pune University, Pune. Her research journey in the field of osteoporosis started in 2008 after joining the department as assistant professor. Along with collaborations from Helberg University, Germany, she is actively working on phytochemicals that reciprocally modulate the gene expression of different bone cells. Her lab is also exploring diversity in the human skin microbiome of genetically related and unrelated individuals of Indian origin, with an intention of using skin microbiome as a complementary system for human identification. This project proposal was selected for funding under Innovation Driven Technologies for Forensic Security and Safety theme of SPARC MHRD Government of India. With this, well, there are many more achievements of Madam, which I am sure will reflect through her talk. With this, may I request Dr. Richa Ashma to proceed with her presentation.
Thank you very much, madam. Am I audible?
Yes.
Yes, madam.
Okay. Thank you very much, Madam Rashmi, Madam, for this nice introduction. A very good morning to everyone, Rashmi, Madam, Khandagle, sir. And before starting my talk, I would like to thank the organizers for this faculty development program for taking the initiative in this pandemic, for organizing the faculty development program in the online mode. This will definitely is going to be very relevant to all the teachers from chemistry and biological sciences background. So with this, I would like to start my talk. So I will be sharing my presentation. Just a minute. Is my slide visible to everyone?
Yes, madam. Slide is visible.
Yes, ma'am.
Okay. So I then need to put it on the slideshow mode, I believe. Now in the slideshow mode, is it visible?
Yes, madam.
Yes, ma'am.
Yes, ma'am.
Okay. So, I think there is some
Madam, can you share it in full screen mode, please?
Yes, ma'am. Yes, ma'am. I will. I will. Now, is this fine?
Yes, now it's
Yes,
okay. Could you kindly just go on to the next so that we can see it moves properly? Next slide and again back. Yeah, yes.
Is it changing?
Yes.
Okay. So now I think I can start my talk.
Yes, please.
So I think that the participants are mostly from the biological sciences and from the chemistry background. Am I right?
Yes.
Or all of them are from the biology background?
No, ma'am. It's around 50% from chemistry and 50% from biology.
Okay. Okay. So I will keep on briefing about, if I will go more in detail about the biological sciences, I will just make it little easier so that chemistry people can also understand. And I believe since I am going to give the lecture on the forensic sciences, it would be of interest for all of them. Just to start with, I'm asking this question, have anyone, like obviously in the pre-COVID era, how many of you have gone to the crowded places like this, to any stadium or to any place which is very, very, very much crowded? So this obviously we are not going to see after this COVID pandemic. So when we go to this kind of a stadium or any place, and if, say, for example, if some kind of disaster happens, then how you will identify an individual? It becomes really very difficult. And we become or we are known or identified only by our ticket number. So we lose our identity. But what forensic science says that if even a single person out of this crowd is unique and different at the DNA level, maybe morphologically the person can be same, the person can have the same eye color or hair color or same kind of height or even the same blood group. But at DNA level, each and every individual is unique. So this is the theme of today's talk: DNA fingerprinting, trends and challenges. So just to start with, can anyone among the audience would like to share their views on forensic science, or do they want to discuss any recent case which they have come across? Anyone? Just to make the session little interactive. Any recent case?
Ma'am, it's the mother and daughter where the mother was suspected to be the murderer of her daughter.
Yeah. So what happened to that case? Like how that case, has that case been solved? Or just by the people's information, it is?
By the forensic science, they could find out.
That the blood on the arm is matching with the daughter's blood?
Yes.
Okay, so these are the kind of tests that forensic scientists do every now and then, you can call it, and they try to solve the mystery behind the case. So this case was very obvious, because mother was there and the child was there, and the people around the colony were knowing that this has happened. but sometimes forensic people get a very difficult situation also. So I want to start with that in forensics, to identify an individual, we consider both intrinsic and extrinsic factors to identify. And in intrinsic factor are those which does not change, they remain unchanged. Whereas extrinsic factor, as the name suggests, it changes with time. So say, for example, it can be further divided into biotic and abiotic cases. So abiotic, the clothing of a person, the ornaments, the cigarette butt if a person is using cigarette, or the vehicle. So you can very well see that these are the factors which always change. It also includes some of the biotic factor like skin microbiome or metabolome, or the insects, or even the habitat of the person. So these come under the extrinsic factor. People nowadays do not use these factors for the human identification. We basically, all the forensic scientists of today's world are focusing only on the intrinsic factor. That remains unchanged. And for biological identification, mainly two different types of factors are considered. One called as the structural or morphological, which also includes all the anatomical, histological, and cellular components. And in this category, fingerprints and body characteristics come into play. And there is another classification which says that biological identification can be done by at the molecular level using the proteins or the nucleic acid. So if you look into any of the forensic cases, not again a single forensic case has identified the convict or the victim using only these morphological features. They always, what they do is they mix these structural or morphological feature with some other factors which are listed here. So say, for example, they mix features with the DNA, or features with the voice, or features with the handwriting, like that, or features with some of the biological or biochemical characteristics. So this suggests that a single or a group of markers are employed to achieve a conclusive identification. And this also suggests that to come to a conclusion, use of molecular markers is indispensable. And this is more evident if you have, I believe that all of the attended audience is of my age group. So all of us have seen this movie, Khoon Bhari Maang, I believe. So here you can see that there was a transformation in the face of this victim due to the plastic surgery. So this is the reason why morphological features are not reliable, because nowadays not only the victim but even the convict, what they can do is they can undergo the plastic surgery and they can modify their features. They can even change their jaw lines so that it becomes very difficult to identify. Now, other factors also like they can change their hair color, they can change they can use a different type of contact lens to hide their identity. So these things are now very common. If you can give some other example related to this, if any one of you have come across any case.
Hello. May I?
Yeah.
The biological parenting case that is very famous in a political background of Mr. N.D. Tiwari.
Yes. So that case also got solved by using the DNA.
Yeah. I think CDFD has
done that.
A crucial role in that.
Yes. Hyderabad, yeah. Hyderabad lab has done that. Mm-hmm. Yeah, and when we talk about this kind of morphological features, or even if you can see like fingerprints, if someone is using gloves while committing a crime, it becomes very difficult to trace that person. So what I want to emphasize over here that biological identification only based on these morphological features is not enough. So from now onwards, in next of series of or time of my talk will be purely focused on the molecular markers. We will not discuss anything related to these morphological or structural features. So if we talk about the history of molecular markers in forensics, the blood group marker, which was called as the first generation marker, was established in the year 1931. And this was followed by the use of protein markers, and nowadays people know that ABO blood group marker or any protein marker, say for example HLA marker, these are not sufficient to identify an individual. If we try to make the profile of individual who are in the audience, you may find there are 10 or 15 people who might have the B plus or B positive blood group. So these markers are not very much polymorphic, and therefore came the second generation marker, which was proposed by Dr. Elik Zaffry in the year 1984. And he proposed that there are variable number of tandem repeats in the genome, and those can be used for the human identification. And the methodology that he adopted was Southern blotting and RFLP based. So these VNTR markers and the third generation marker, which are STRs and the SNPs, they belong to the non-coding region of the human genome. So I need not to tell you that the human genome, even for the chemistry people, they might be knowing that the entire human genome has two components. Basically, you can say that one part codes for a particular expressed region or codes for a gene, whereas the other part is the non-coding region, which does not express for any gene. And these non-coding regions have the repetitive sequences also, which again can be of 2 or 3 different types. So one type is the tandem repeat. Let's say, for example, and in this tandem repeat category, there are again 2 different types, mini satellites and the microsatellites. The microsatellites and these micro and mini satellites are mostly located near the centromere of the chromosome. The mini satellites are the VNTRs, or the variable number of tandem repeats. So, and the number of nucleotide which gets repeated tandemly is around, say for example, 12 or 13 nucleotides stretch you'll find, and that gets randomly repeated in the genome. Whereas the microsatellites are small or short tandem repeats, like say for example a dinucleotide repeat units or a trinucleotide repeat, tetra, penta, even 7 nucleotide repeats has been seen in the genome near the centromere region. So what Elik Zaffry did is he used the VNTR markers, and but then nowadays it has been found that this is even not sufficient. I will come to this point again. Then came the third generation, which uses the STR markers or the short tandem repeats, and the SNPs, which are single nucleotide polymorphism. So why there was a transition in the marker is Elik Zaffry suggested that whenever you're selecting a marker, it has to be polymorphic. It has to be very fast evolving. And there has to have a codominance inheritance pattern of the molecular marker. Why codominance? Is because, say for example, if we go by, you must have heard about the Mendelian hypothesis, Mendelian theory, that one particular allele becomes dominant over the other. Then in F1 generation and even in the F2 generation, you may find a predominance of the dominant allele. So that should not happen. Otherwise, what will happen? The profile of any individual will get influenced by the parent. So What he suggested is whenever you are working on any molecular marker for forensic purpose, that should be codominant in inheritance. So even the VNTRs, even the ABO blood group, you might be knowing that they are the codominant, they follow the codominance. And the third point, what he suggested is that they should be randomly and frequently present in the genome. It shouldn't happen that they are restricted to a particular place in the genome. And that's why whenever we see all these microsatellites are very randomly and very rampantly present in the human genome. They are reproducible, meaning if a particular lab has given a profile of an individual, you will get the similar profile if, say, for example, you are doing it in the Hyderabad, or say, for example, you are doing it in the Germany. So lab to lab, there should not be any variation in the result. The fifth point is it should be easy, doable hona chahiye, and it must be cost effective. So these are the features that he suggested. Now I would, because Alex Zaffrey is also called as the father, or he is the pioneer of this forensic science, and he only provided the VNTR markers. So I would like to give a little pause on his work. So as I said, he was working on the second generation marker. He is Alex Zaffrey. So how he was working on these VNTR markers for a very long time. And when he was working, a case of Colin Pitchfork came into existence. Like, he was the person who victimized two teenage girls. He raped them and finally he murdered them. So when Alex Zaffrey started working on this case, he found that VNTRs are the best marker which can identify a person. And because of his dedication and work on VNTRs, Colin became the first person to be convicted using DNA profiling. And this happened in the year 1988 when he was sentenced for life imprisonment. So this second generation marker gave a big boost to the forensic scientists, that DNA can be used for the identification of the human. And I have already discussed the features forensic marker features which a forensic marker should have. So again, it is listed over here. So what I want to show you over here is a small video. This is a second generation marker, and this is the conventional way of doing the fingerprinting. But this is not the method nowadays used by forensic scientists, because people are now using the third generation marker. But what I feel is in many schools and colleges, teachers are still using this way of DNA fingerprinting. I will show you the video and you can very well correlate if they are explaining the similar technology or not.
Obtaining a genetic fingerprint.
Is this video visible?
Yes, madam. Yes.
Yes,
Okay.
madam.
Okay. Begins with a sample of biological tissue. At Guy's Hospital in London, this research worker is using an automatic DNA extractor to produce a high-purity DNA fraction from a blood sample. A restriction enzyme is added to the DNA. This enzyme moves along the DNA strand, cutting it at the so-called restriction sites. The restriction sites available to enzymes are located throughout the chromosome, except in sections consisting of copies of the core sequence. This process produces many pieces of DNA, which consist of repeats of the core sequence. The mixture of these DNA fragments is then placed onto an agarose gel. A small voltage is applied across the gel, and the fragments move through the gel at a rate proportional to their size. Because gels are difficult to handle, the DNA band pattern on the gel is transferred to a nylon membrane by a technique known as southern blotting. First, the nylon membrane is placed on the agarose gel. Then, absorbent paper is layered on top of the membrane. It acts as a wick, drawing up the DNA bands onto the membrane where they stick. Finally, the researcher adds a radioactive DNA probe which will bind specifically to the core sequence. Excess DNA probe is washed off, leaving only the radioactive probe that is bound to the DNA pattern on the membrane. This can be shown on X-ray film, producing the genetic fingerprint.
So, anyone has any question till this part of the talk? Anyone would like to comment on this technique? Hello, am I audible?
Southern blotting.
Yeah?
Southern blotting.
You want me to explain that?
No, is this technique of southern blotting?
Yes. That is the technique of southern blotting. Like, it helps in transferring whatever is bands have come on the gel to the nitrocellulose membrane. And then that membrane is developed and pattern can be preserved on it, because gels otherwise cannot be preserved and cannot be put forward for any case in the court. So we do the southern blotting, and then the pictures are restored for any case which is being handled. So any question till here?
Ma'am, but nowadays this is replaced, am I right?
Yes. Nowadays it is replaced, and because of these advantages which are listed on the screen, because it is expensive and too much time consuming and very labor intensive. You have seen that the person has to continuously work on the table for the wet lab, and after doing so much of labor, the throughput is very low. Like, you can do only one locus at a time. And the locus, say for example, VNTR, if we will look into the number of alleles, the VNTRs have very low allele number. So we can call it as a less sensitive marker compared to the third generation marker, which is the STR. And as I said, that the VNTRs are variable number of random repeats. That is the acronym. So the repeat units are very long. So when you have to amplify a longer sequence and all the alleles of the Langmuir sequence, obviously you will require a large sample size, and that too, the sample or the blood sample which is normally obtained from the crime scene are not that in good condition. So the requirement of this VNTR is too high to meet in a forensic case. Okay, so these are some of the shortcomings which is there in this system, or the use of VNTR or the second generation marker. Okay. So and this was, as I said, this was being used in 80s and 90s. So since then, forensic science has undergone a very fast evolution. And so our talk will be focused on the third generation marker of forensic science, that is the STR and the SNPs. So in this talk, we will cover not only the timeline which is mentioned over here, but also about details about the markers which are used, that is the STR and SNPs, how the workflow is done. You have seen one video on the second generation workflow. We will discuss about the workflow of the third generation marker. What are the advancement that has been made in the technology, and followed by the databases. So is this workflow is fine with everyone? Or anything else you want me to include?
No, ma'am, it's fine.
Okay. So if we look into the forensic timeline, so after the introduction of the VNTR marker, which was in 80s, differential extraction came into picture. So what is this differential extraction? It is started, as it is written over here, it is started in 1985. But this helped in identification of male and female component or DNA characterization of male and female characteristic. So this helped in solving rape cases where most of the time we get a mixed sample of male and female cells. So this is, if you call it, this is also a one landmark in the field of forensic science. And then came almost in late 90s, the mixture analysis or the employment of different VNTRs, not single VNTR, 4 or 5 different VNTRs were started being used for identification of the human being. And in 1998 to 2000, this is the turning point when CODIS loci, I will come to this CODIS loci again, but these are the STR loci, the third generation is started. CODIS is combined DNA indexing system, and this was introduced by US, the FBI, Federal Bureau of Investigation of US, introduced these CODIS loci, and they made mandatory for all the forensic labs over the world to follow these CODIS loci, STR markers, to profile an individual. And during the same time, automated capillary electrophorensic system was introduced by ABI in the market. So this made the profiling of this CODIS loci easier for the forensic labs. Side by side, people started, when this was introduced, side by side, people started making the databases. So CODIS database as well as the Y chromosome STR database was launched in the year 2005. But again, people observed that this is not going to suffice the need of the cases when the amount of DNA is very limited. So for low copy number condition, they introduced the probabilistic genotype software, which can interpret the result and which can probably, you can say, it can, what to say, like it can tell you like this is the most probable genotype of this particular person. So people started working on the development of this software, and US validated it in 2015, UK validated it in 2018. So since 2018 to 2021, what people are doing now is they are now focusing on the coding region of the genome. And they are trying to see if that part of the genome can also be included for human identification. So this is the forensic timeline. And you can see it has evolved very fast. So since we will be focusing on the third generation, so let us discuss more about the third generation marker, which is the STR or the short tandem repeats. You can call it microsatellites also. So as I said, microsatellites can be the tandem repeats of di or tri or tetra or pentanucleotide repeats. So this is how it gets repeated. So say for example, every unit is, say for example, tetranucleotide. So it gets repeated multiple times. So when we try to amplify this region of the genome, we have to use the primers. And the primers are designed in the flanking region. So if, say for example, if a person, obviously we are diploid, so there will be 2 alleles. So on one particular, we have the allele 1, say for example, has 8 alleles, and the other 8 repeats, sorry, and the other allele has, say, for example, 7 repeats, then the genotype of this particular person for this locus will be 7 and 8. So the way we have perceived genotyping, say, for example, we have, since our childhood, we were saying that for tall, we will be either capital T, or capital T, or small t. So that is the perception of the genotype we have. But here, for STRs, it is always in numbers. This is the difference. So if the person is homozygous, then it can have the 7 repeat units. Or if this person is, say, for example, heterozygous, then the number will be 7-8 or 8-7 or 7-6 or 7-5, like that. Or even 5-6. It's not that the 7 number will remain constant. So one is coming from the father and the other allele is coming from the mother, and both are codominant. So both will get expressed in the genotype of the individual. So I hope this is clear to all of you.
Yes, madam.
Yes, ma'am.
Okay. So as I said that the STRs are polymorphic, highly polymorphic, and that was the basic feature that a marker should have. So FBI in US introduced the concept of CODIS, and it is a set of 13 STR loci. Loci is what? Loci is, or locus, this is the plural form. Locus is a region in the genome where these particular STRs are located. So 13 CODIS loci was introduced by the FBI of US, and why these 13 CODIS loci is purely because they are located on different chromosomes. They are mutually independent while being inherited to the next generation. So every lab over the world has to use these 13 CODIS loci for profiling an individual. One reason behind selecting these CODIS loci is because they have a defined size range. So say, for example, as I said, that VNTRs require a large amount of sample in a good quality and quantity. So these STRs, they require a size range of, say, for example, 100 to 500 bases pair, which any forensic sample can have at least this of intact length of the DNA. The third point, which is again very important, is all these 13 loci can be multiplexed. Multiplex means what? All these 13 loci can be amplified or PCR amplified in a single tube. PCR, I hope all of you understand. Polymerase chain reaction.
Yes, ma'am.
Yes. Even the chemistry people. Right? So,
Yes, ma'am.
Okay. So this is these 13 set of loci can undergo simultaneous amplification. Fourth very important point is the profile of a person can not only be stored in this database, but also can be compared. So you might have seen in the detective serials that they have put in the profile and it comes on the screen, match found. but let me tell you that is now not how it works, but they portray it like that. So this particular database is used to compare and store the genotype for identification of every individual in future. And this system is so robust that the odds of finding two individuals with the same set of 13 STR genotype is 1 in billion. And the total world population is also in billion. So you can say that this 13 loci is good enough to identify an individual out of a group of billion people. So how it is possible? So here what I have done is I have listed all these loci. And you can see that the number of alleles which are there for these loci. So you can imagine ABO blood group. ABO has, say, for example, 4 different alleles. So Compared to 4, each and every loci which are listed here have more number of alleles. And it is said that markers with maximum number of alleles are more suitable for forensic identification. And along with these markers, amylogenin gene is used to identify the gender of a person. Okay, so what makes this system robust is these highly polymorphic locus. So I just kept this slide keeping in mind the people from chemistry background. So this is how the polymerase chain reaction works. It has 3 steps. Since I have already asked that all of you understand, so I will not go into the details of this. And you also know that a small region of DNA, when gets amplified using the PCR, the copy of this particular template gets amplified in exponential manner. So say, for example, by 35 cycles of this PCR, this small DNA fragment will get amplified into 68 billion copies. So this is the robustness of the technique. So let us now start with the workflow of the DNA fingerprinting using the 3rd generation marker. So it starts again with the biological sample collection from any crime scene, followed by the DNA extraction, its quantitation, then the PCR multiplication. Obviously, the multiplexing is done, so multiple strands are there in a single tube. Once the amplification has happened, we subject that amplified DNA to the genetic analyzer. This genetic analyzer has inbuilt softwares, which is responsible for the STR allele designation and providing the STR genotypes of a sample. So the genotype is displayed as this chromatogram on the screen. And these peaks are the bands, DNA bands in the form of peaks. So it becomes very easy to compare the genotype of an individual in the database. And once this comparison is done, the probability of match and power of discrimination is calculated. And this is also done in an automated manner. So this second part and the third part of the present day DNA fingerprinting is fully automated. What is not automated is this first molecular biology part, where we have to collect the sample, amplify, extract the DNA, and amplify it. So the chances of error in this part of DNA fingerprinting is very high. So what I want is to discuss each and every step in a little more detail. So this is a very famous scene. You can find it anywhere on the Google. So from any crime scene, a forensic scientist can get any kind of these biological sample. So a forensic lab must have a very established and reproducible protocol for DNA isolation from any of these biological material. The problem is not that you have to isolate the DNA from hair shaft or from the bone, tough tissues like bone, and from a very small tissue like hair shaft. But the challenge lies that in most of the cases, these samples are highly contaminated. You might find the samples either with the mud or with some kind of fiber on the carpet, which gets confused, like whether this is hair or the fiber of the carpet. Not only that they are contaminated, they are also present in very small amounts, sometimes in traces. And sometimes, to befool law enforcement agencies, these criminals, what they do is they mix the sample with the non-human sample. So you also have to identify the species, whether it belongs to human or it is a non-human sample. So these are the basic challenges which a forensic scientist encounters with every biological sample. So it is said that isolation of DNA or the extraction of DNA from the biological sample or forensic sample is not a science. It is an art. It requires a lot of patience, a lot of practice. Why I am saying this is the crime scene samples are very precious. It cannot be reproduced if lost or if mishandled or if some contamination happens while collecting the sample by the collector itself. So all our personnel, police personnel are trained for this. They get training for this. They have to collect all the biological samples from the crime scene, whether it belongs to victims or whether it belongs to suspect. They do not have to discriminate among this. They just have to collect the biological sample. And if required, they also have to give their own sample so that the mixing can be ruled out. Even the person who is going to perform the DNA fingerprinting, they also have to contribute their sample. And these all things are done so that they can rule out the mixtures, because mixtures are real challenge and issue in forensic identification. And it arises when 2 or more individuals contribute to the sample being tested. And it becomes really very challenging if the scientist does not have the extensive experience or training of handling these mixtures. And it further becomes complicated if the forensic lab is using low polymorphic loci. Now let me explain you why. You can see the profile over here. We know that human beings are diploid. So if 2 peaks are coming, you will otherwise say that this is not a mixture. But if you will come across the genotype of other loci, you can very well say that some kind of mixture has happened over here, because a single person cannot have these many alleles, right? Likewise over here. So if you are using a large number of loci and polymorphic loci, it becomes very easy to understand that mixture has happened or it has not happened. Otherwise, if less polymorphic loci are being used, and that too in 1 or 2 loci are being used, if not 13, then the result will always remain inconclusive. And you will just remain ignorant about the mixture that has happened. So DNA fingerprinting is done very carefully. This is how the allele designation is carried out. I have told you that it is done by the inbuilt software, which is the GeneScan and the genotyper. This comes along with the machine, ABI machine, the automated DNA sequencer, which converts the base pair, DNA base pairs, into the genotype using the allelic ladder. Now this allelic ladder comes with the STR kit, which is used for the CODIS loci, and it covers all commonly found alleles in the world population. So say, for example, I just want to give you an example. Like, say, for example, if a person has 2 peaks, by 2 alleles will be there in a person. So say, for example, for this particular locus, D3S1358, if a person is heterozygous for this particular locus, and if the bands are at 110 and 120 base pair, then the genotype of the person will be, you have to just extrapolate these 2 bands over here, and the genotype of the person will be, say, for example, 12 or 15. And that is how it is done for rest of the markers. So, and it is done by the automated or the softwares which are inbuilt in the machine. Now come to the last part, which is how this particular discrimination power or matching probability is calculated. So this is based on the concept of probability. If, say, for example, we are using 9 locus, okay, 9 locus. So the probability of finding the same genotype which the individual has will be 1 multiplied by all the number of alleles which a particular locus will have. If it is not clear to you, please let me know, because this is the basic concept behind the entire third generation.
Ma'am, if you don't mind, could you please read it?
Okay, so you remember this particular slide wherein I have shown all the, right? So say for example, you are using loci for identifying an individual, right? Every individual has 2 alleles, so the genotype will be a combination of all these alleles which are there. So if you are working on one particular locus and the locus has, say for example, 9 alleles over here, D3S1358. If 9 alleles are there, then the genotype will have 18 because everyone is diploid, right? So this 18 will multiply it with the number of alleles which are there in the next locus. So TO1, if it has 10 alleles, then 18 into 20. It works on the multiplication rule. Then you have the next marker, D21S11, which say for example has 25 alleles. Then the genotype of the person might have the 50 because they are diploid. So this will get multiplied each time. So at the end of genotyping of 4 markers, you will be able to identify a particular individual out of 7,92,000 individuals. Are you able to understand this?
Yes, ma'am.
So now you know that CODIS, which is based on 13 loci, so how powerful that multiplication might have given the result in identifying an individual. So with 9 loci, you can come closer to the billion. 100 billion ho jata hai. And today's world population is how much? 7.8 billion. So using that 13 loci will be sufficient to discriminate a person from the entire world population. You can easily identify them. And you will not believe, the forensic science has not stopped over there. They are now using 24 loci system. I will come to this slide again. So in continuation of that discrimination power of the CODIS loci, what people have done is they have compared the matching probability using different sets of loci. So initially people were using 8 STR markers only, and they found that the matching probability is this one for different population. These are the different population: Hispanics, Caucasian, African, Asians, etc. And as they increase the number of markers from 8 to 15, the matching probability decreased significantly. And what this suggests is, as you will keep on increasing the number of loci or the markers for forensic identification, the matching probability will keep on decreasing. And you can very well discriminate one individual out of the entire world population. And this is the power of the today's forensic identification, human identification system. And I said that nowadays people are using 24 loci. So you can very well imagine it's not just the extrapolation of this particular data to 24x. They have really calculated for all the population, because they have to sell their kits in the market. So people are working on these 24 loci, and which otherwise, if you will use a normal gel, it becomes very difficult to make out which particular band belongs to which loci. So this is the beauty of today's technology, that with the help of automated sequencer, you can look into these base pairs as DNA bands in the form of these peaks, which are easily interpretable, very user-friendly. So this 24-plex system uses 6-die chemistry, meaning that the primers of these 24 loci are labeled with 6 different color fluorescents. And all these dyes have the non-overlapping excitation and emission wavelength. And they have particular filter for this in the system, ABI automated DNA sequencer. So all these 6 dye chemistry filters are available in the system, with the help of which you can very easily identify the peaks of these 24 loci. So I can very confidently say that the system present in today's forensic labs are very robust, very time efficient, and very cost effective, because in one run, you are identifying 24 locus at a time, and the complete profile of the individual is available, say for example, in 3-4 hours. And the system robustness, I have already shown you that how robust is the system. It can identify one individual out of world population. So till now, I was talking only about the 3rd generation marker using the biparental DNA. So both the parents were contributing to the profile of an individual. And as I already said, that these are in the non-expressed part of the genome. What forensic scientists do further is they use the lineage markers also. Lineage markers are what? They are maternal. They use maternal DNAs also and paternal DNA also. You might have heard about the paternal testing, or if you want to identify somebody's mother. So maternal DNA is contributed by the mitochondria. We all know that about it. And these mitochondrial DNA are used only when the nuclear DNA is not available for testing. Sometimes this kind of situation happens when the DNA is very old or very dead. In those circumstances, you have to bank on the mitochondrial DNA. You might remember Netaji's case, where we got only the tooth. So from that tooth, and that was so very old that it was very difficult to amplify the nuclear DNA samples, loci. So in those cases, we bank on the mitochondrial DNA, because they have a very high copy number in each and every cell. Mitochondria are present in large numbers in every cell, and that suffice for the amplification if nuclear DNAs are not available. So, and the inheritance pattern you can see over here, that every mother contributes mitochondrial DNA to the children. If the mother is different, again the profile will be different. So this is how it is transmitted to the next generation. Then we have another marker, which is from the Y chromosome, that is used for the paternal DNA testing. And if you will look at the inheritance pattern of this, it is always from father to son, and from son to the next generation son. So these lineage markers, they have a different type of inheritance compared to the autosomal marker, where both the parents were contributing to the genotype of the individual. So any question till now here?
No, ma'am.
We can proceed?
Yes, ma'am.
So talking about these lineage markers, they have some advantages and disadvantages. So advantages includes that they become very useful in tracing a person beyond a generation. So say for example, if Saddam Hussein is hiding somewhere, and if you come to know about his lineage person, you can very well trace that this person belongs to the lineage of Saddam Hussein. Or see, for example, in mass disaster cases, this in 9/11 हुआ था. So 9/11 में most people were identified using these lineage markers, the Y chromosome. So missing person cases, when you have to trace this person from beyond a single generation, these lineage markers becomes advantageous and we go for them. But the main drawback of these markers is they have very low power of discrimination because there is no recognition taking place. So they do not evolve. They are not fast evolving marker. Okay. And if you compare one family member with the other, they become indistinguishable. Because they also have the same YSTR the father has. Right? It has not undergone any recombination. Likewise, mitochondrial DNA. They do not undergo any recombination. So the family members, you cannot distinguish. But advantages, as I said, and further to emphasize, the YSTR becomes especially very important where we have to do the sexual assault cases and when the multiple males are involved in the cases. So look at this particular electrophotogram. So otherwise, when you have a single person involved, you will get single peak for all the YSTR markers. The moment you get two peaks, because Y chromosome cannot be present in two copies, right? So the person will have only one peak in the normal circumstances. But if in any sample you are getting two peaks, it means there are multiple males, or say for example 3 peaks, then multiple males are involved in that case. So this visual interpretation, which is so very easy, makes the case very simplified. And you can, in minutes, you can say that no, this is not a normal rape. Multiple males are involved in this case. So easy. Now, almost we are coming to an end. This picture is just to show you or sensitize you about the use of coding regions, which has recently been started in the forensic science. Initially, when this started, people were using less number of locus. So they were not able to make out the complete picture of a person. This is just an analogical, I'm just trying to give you an analogy. So say, for example, with the help of coding region markers or the region which gets expressed, if you are using them, and if you are using the less number of such genes, you will not be able to profile the entire face of an individual. But if you increase the number of puzzle pieces, if you explore more of them, then, and if you are, say, for example, using 40, a little more clearance about the face, whether it is oval or whether it is oblate, kind of thing can come into picture, and it becomes certainly very easy for the forensic scientist to recognize the person. So this kind of thing is called as the DNA phenotyping, because we are trying to make the morphology of the person using the DNA typing. So let us discuss more about it. What people are doing nowadays is they are using the irisplex to see or to explore the eye color of a person for identification. The second type of testing which is being carried out is called as the irisplex, where iris and hair color is being used for the identification. And then the third part, which is DNA chip multi-express gene test, in which they have also included the skin color. So all the variations in the shades of iris or hair or the skin is, people are trying to cover the entire variants which are there. And the database which has been generated based on this testing, these 3 testing, have certainly right now they have the low number of individuals, but it is going up. What I want to emphasize over here is the 3rd generation marker. Till now we were discussing about the STR, but in this expressed region profiling, they are using the SNPs, the single nucleotide polymorphism, which theoretically is beneficial due to small size. They are present at single nucleotide position, so amplification becomes very easy. And for this purpose, they have till now profiled around 41 SNPs to cover hair color, iris, and skin. S is for the skin system. And they are covering many more genes. I have just listed 3 over here, but you can go to the database and you can see what are the different genes which are involved. But what I want to emphasize here is these SNPs, however, will not be able to replace the STRs. So STR and SNP together are part of 3rd generation. It's not that the SNP will make a 4th generation marker. So this is because the STR database, which is right now present for the world population, is much more informative than the database which is for the SNPs. The STRs, as I also said, that can be used for the mixture detection, where more than one individual has contributed into the crime. That mixture analysis cannot be solved by using the SNP. So this is one major drawback of using, if a person is using only SNP. The third is the degraded DNA can be successfully analyzed by the use of mini STRs, where we have further reduced the size from 100 to 500 base pair. It has been further reduced to 100 to 250 base pairs. So such system is not yet available for the SNP. So we can say that SNPs are not going to replace the STR. They are certainly going to complement the STRs. So,
Madam, sorry to interrupt you. I have a question or a doubt. Madam, nowadays we have the Aadhaar, the unique identification system. While getting the Aadhaar, they takes our biometric data like fingerprint and iris and all that. So, is there any a set of alleles or genes that are actually related with this kind of identification, like biometric? Each and every individual is having a unique fingerprints.
Yeah.
So, what you can say about that, Miss? Is there any report that certain a set of alleles are actually giving expressions for the fingerprints and all that?
Yeah. So as I said, like people are becoming very smart day by day. So they tend to change their hair color or eye color by using a different color contact lenses, or even hair color by using certain dyes. So law enforcement agencies, they know that if they will use only the morphological features, the convict may escape. So what they are doing is they are using the DNA markers for this. And I have listed some of the genes which are being used for analyzing the SNPs which have been found for these phenotypes. So say, for example, recently I have communicated one paper on MC1R gene. So this MC1R gene is basically for the skin color. So there are variants, SNP variants, which are available around the world population. So I have done on the Indian population, and that too I have restricted myself only to the Maharashtrian population. We have taken 8 communities. 4 were from the tribal areas and 4 were from the urban areas. What we have found is there are certain SNPs which are predominant alleles of that particular SNP is predominant in the area. So that helps us in understanding, and we have also done the melanin indexing of the individual, of the same individual, and then we have done the genotyping. So we are trying to correlate over there that if, say for example, the melanin index of an individual is, say for example, towards the darker. I'm not giving you the figures because it becomes difficult to understand. So say for example, if the melanin index is high or the person has a dark complexion, then what particular allele of the SNP is predominant in such people? So even if a person changes, bleaches himself or herself, that SNP allele is not going to change. Even if a person is using a colored contact lens, but that SNP is not going to change. So if you have profiled a person based on these SNP, and if it does not matches with the phenotype, you have to have a faith on the DNA data first. So it is becoming day by day robust, and people are working day and daylight, you can call it, on these markers to finally conclude that there is a correlation between the skin melanin index and, say for example, one particular SCR1 SNP. I can name it, but it is of no use because the similar SNP is not there in the European population. So there is a variation, and because the paper is under communication, I am not revealing the entire data to you, but very soon you will get this kind of data. And I will show you in the data, I'm going to show you the database over here. This is for the database. You can also go to the SNP database from here. So is my answer then or explains the confusion?
There is another question from Ms. Megha.
Yeah.
Yes, ma'am. Can you learn?
Yeah. Then.
Relation found between the eye color, hair color, and skin color. I mean, is there any relation, like a particular eye color will have a particular skin color? Is there any interrelation?
No, not yet. That kind of study has been carried out. Right now, we are doing individual phenotype studies. Like people are working on eye color independently, and they have not discussed and they have not used the same essence or the marker or the gene for the other phenotypes. Say, for example, for the hair color. So that correlation study, that's why it has not been come into the public domain. It is under preparation, kind of, you can call it.
Yes. Thank you, ma'am.
Yeah. Any other question? No? So there, I said that there are STR databases, and we were just now discussing about the SNPs. So the database which is based on the SNP is called as the forensic chip, and today's SNP database includes the autosomal SNPs, X chromosome SNPs, the Y chromosome SNPs, also the mitochondrial DNA SNPs. So the total number of data right now we have available is around 2,01,173, and this gets updated very frequently. So in year 2021, version 5 is available. Both the STR database and the SNP database is not a public database like NCBI. NCBI is a public database, but the forensic databases are not public databases. It is available only to the law enforcement agencies. And I want to say over here that the STR database was created by John Butler, and the database is used very frequently in all the different cases, say for example, for mass disaster cases, for missing person identification, for if any kind of military casualty has happened, they refer to that database. Then for the parentage testing, then even for day-to-day genealogical studies, or if someone wants to authenticate any human cell line, then you have to profile the entire codis loci and say that it belongs to the human. And many more, like say for example, for genetic diseases, also the mapping has to be carried out. And we do also the population-based study. If you want to do the diversity analysis, as I said, that we have considered a tribal population and the urban population, and we are trying to. So this kind of population-based study also is feasible using the databases of the loci or the STRs. So these databases, further, I want to say that are so robust and so searchable, and sharing of these DNA databases is also done, provided there is an agreement or treaties between two countries. Otherwise, that particular country will not be able to access the global data of the DNA file. Searching can also be done for the presence of a matching profile. So any data when is deposited, it is first searched for the matching profile. If it is already available, then the database will reject it. If it is not present, then the profile is accepted. So this is how it works. So DNA profile of a wanted criminal is searched on the Interpol database based on the international guidelines. So because this is such a sensitive data, it is not available for the public. And it is mandatory for all the world population, for all the forensic labs of the world to cover a minimum 13 codis loci. Otherwise, they do not accept the profile of the individual. So, what at the end I would like to say is, if you will look at these guidelines, you may feel that FBI US has made the guidelines so stringent that they say that you have to have a rapid DNA testing for the international forensic laboratories. It has to be fully automated, meaning hand-free system needs to be used for profiling the codis loci. And they also say that if you want to deposit the data of a normal individual, not a criminal, normal individual, the sample has to be the buccal swab, meaning non-invasive method has to be used for the collection of the sample. Concept behind this is, what they say is swab in and profile out. मतलब आप swab को डालो system में, and what you will get is the profile out. There should not be any manual intervention in the profiling. It has to be fully automated, whether it is DNA extraction, amplification, or detection, or allele calling. All these steps has to be fully automated. So this is all about the third generation forensic technique which is followed. What are the challenges? I told you, like in some of the laboratories, the full automation has not happened, but that is the expected from every forensic labs. And what are the new technologies which are going to come? That also I told you. So is my time over, madam?
Almost, madam, but we have 10 minutes more. We can keep it for the question answer session.
Okay.
Or you can extend 5 minutes more.
Okay, so I just wanted to sensitize the audience, like what we are doing, why I am giving this talk, because I am also a zoology associate professor in Savitribai Phule. I do not work on any forensic lab, but what I am working on is on this Locard principle, which says that every contact leaves a trace. So what we are using, how we are using this Locard principle is we are doing skin microbial profiling. So mainly you can say, apart from melanin indexing and SNP studies population, we are also performing this skin microbial profiling. And we are trying to see that if at places where biological evidences are not there, so rather than getting nothing from the crime scene, if we can use these skin microbial profile for human identification, then both these will complement and confirm the identification of an individual and can put forward the conclusive evidence in front of the court to after identification. Because some of the loci which we use, they really require large quantity of DNA. And when the samples are degraded, you do not get the complete profile. It always remains partial. And partial result, as we have seen, remains inconclusive. So if in such cases, if we can include the skin microbial profile. So there was a paper in 2010 which suggested that when a person touches a surface, it shares the microflora. And this paper says that the 13% of the bacterial phenotype are shared between the two individuals. So we derived one conclusion from this statement, that meaning rest 87% is with the same individual, remains with the individual. So if that 87%, can it be used for the identification of the person? So is that 87% is the signature of that person? So the very signature cannot be copied. Likewise, it cannot be transferred. And if you people are interested, you might have heard about the human skin microbiome project which is going on. And what they have done, they have classified the microbiome into two parts. They have called some of the microbiome as the resident and the other ones as transient. So transients are those which gets transferred. So this 13%. But there are certain bacteria which remains on your body surface. They are called as the resident. And what they have also done is they have classified these resident and transients based on their biological site on the skin. So some of the bacteria, say for example on your dry surface of the skin, you will find proteobacteria as predominant one. If the surface is moist, you will find more of Staphylococcus and proteobacteria. So likewise, and because this is dependent on so many factors which are listed here, so we can say that every individual has a very different set of microbiome on its skin. And this we are trying to correlate with the odor of an individual. And you know that odor of every individual is different. And that is being used by the law enforcement bodies since time immemorial. Because sniffer dog ka aapko pata hoga ki they use those dogs to trace the individual. And this human odor is because of the bacteria which acts on the sweat. Sweat otherwise is odorless when it is secreted by the gland. So this odorless secretion of the gland is a precursor molecule for these bacterial enzymes. So these bacterial enzyme converts these precursor molecule into a human odor, which again is governed by different factors like age, or say for example menstrual cycle, or by the emotional factor, hygiene, cosmetics, everything. We know that if a person is a non-vegetarian, or if a person is using more of garlic or onion, they have a different odor than a vegetarian or a Jain person. So it is said that the presence of a particular or the relative abundance of a particular volatile compound gives the human odor. This was published also in 2011. But stating and we published also that not only the relative abundance, but also the presence or absence of a particular VOC, volatile organic compound, defines the chemical profile of an individual or odor profile of an individual. So we are doing this kind of work with the collaboration of our Finland University under the SPARK. SPARK is a very flagship program, if you can call it, of Indian government. So with the help of people from University of Turku, Finland, we are doing this work. We also have a hand-holding with Professor Meeta Ravi Kumar. She is a professor at IBB at SPPU, and with this group we are doing this kind of work, and we are trying to contribute little bit to this forensic science. I'm not very sure whether we will get a positive result or not, but we are trying to prove that there is a microbial signature in every individual, and every individual odor is unique, is known. So something, if it can help out in the forensic science. So with this, I would like to thank the organizers for giving me opportunity to talk on the topic. Rashmi Madam, Khandagle sir, I'm really very thankful. Thank you very much.
Thank you, ma'am. Is there any question from participant?
Ma'am, yeah, can I stop sharing and turn on my video?
Yes.
Yes, ma'am.
Ma'am, my question is, in future, can we relate this microbiome with the crime scenes?
We are trying to prove it. First, we have to establish that every person has a unique signature of microflora. So what we are trying to do, we have taken the individual from, right now we are working only on the Maharashtrian. So we have taken individual, we have identified them, a set of 15 individual we have started with, and we are trying to profile those individual, say for example in summer, winter and monsoon, and trying to see if the same set of microbiome remains on the skin. This is one way of doing it. The other way, what we are doing, even if the person eats, say for example today a vegetarian diet, sometimes if he or she takes a non-vegetarian diet, how much is the variation that is happening? So first we have to prove that it is a signature. It does not change. So for these n number of parameters needs to be tested. We are testing few. I am not very sure whether we will be able to test the entire set of parameters or not, but we are just giving a try.
And how many groups in India are working on this project, ma'am, of microbiome?
Shauchi's group you might have heard.
Yes, ma'am, from NCCS.
Yes, so they are also working on the similar project. Their angle is not the forensic, but they are profiling the individual both for the gut flora and for the skin flora. So they have taken individuals from the tribal belts also. I think anthropology department of SPPU is also involved in that project. Ayush Ministry has given funding to that. And obviously that NCCS is a institute under DBT, so DBT is also giving fund to this project.
Thank you, ma'am.
Any more questions?
Good morning. So I think you said you have taken 15 samples in Maharashtra. So how diversified are the samples?
Sir, first of all, I would like to say that that is not good enough. That is not the representation even of the complete Maharashtrian population. But we are starting the work. So this is just a pilot work which we are doing. We are also standardizing all the protocols and other things. So once those things will established, we will go ahead with more number of samples, because profiling a person for the entire microbiome. So it is not, I said this to Neelam madam that we are trying to do the profiling for different seasons, but at the same time we are also the profiling for different parts of the body. So if we are taking this hand, we are taking the palm, we are taking the antecubital fossa, we are also taking the armpit region. So because what we believe, this surface, because we keep touching different sites, so what happens, it keeps changing. So using this profile for signature is not a good idea. But for forensic purpose, obviously we have to consider this, because every criminal is going to touch the surface using the hand, not from the armpit. So there are multiple factors that we have to standardize and look into it before coming to that conclusion. So we have started the work. Let's see if at all it works. Yeah.
Okay. Many thanks, Dr. Richa Ashma. The session was very interesting and informative. You cleared almost all the concept in DNA fingerprinting. I would like to discuss with you on this topic, as very soon we are moving towards autonomy and we are thinking to introduce few modules and yeah, thank you so much once again. May I now request our participant, Mr. Mangesh Deshpande sir, Department of Microbiology from Hadapsar College, to propose vote of thanks.
Okay. Good morning, everybody. On behalf of all the participants and organizers of this FDP, I would like to express my gratitude towards Dr. Richa Ashma, Associate Professor from Zoology Department of Savitribai Phule Pune University, for giving an interactive talk on DNA fingerprinting, trends and challenges. Madam, you explained the topic in very simple language, giving numerous examples, including a video on southern blotting technique. You elaborated forensic timeline also, in which you gave example which techniques were used in the earlier days and now replaced by modern techniques. You emphasized on third generation techniques and the markers used for profiling purpose. You also explained the workflow of DNA fingerprinting with the examples. Yes, there are many challenges in applying these techniques, but these can be overcome using techniques like gene scan and genotypers. You also explain how matching probability can also be used for profiling purpose. In the end, you also showed how SNPs and forensic chips can be used for solving these problems. I think we understood what Dr. Salunke from one of the TV serials, CID, must be doing in his laboratory to identify the criminals. And it was really a fantastic interaction with you, madam, and thinking of more interactive sessions with you in future. Thank you very much once again.
Thank you so very much for such a nice vote of thanks. And I really want to thank Rashmi madam and Khandagle sir for inviting me for this talk. This topic is really very close to my heart, and I'm ever ready for giving any kind of talk on this topic. So thank you very much. Really, thank you very much. Thanks.
Thank you. Thank you so much.
Thank you so much, ma'am.
Thank you.
We enjoyed it.
Thank you, madam. Thank you.
Participants,
so Rashmi madam, can I log out?
Yes, please. Thank you.
Thank you, ma'am. Thank you.
Participants are requested to remain in the meeting as the next session will start soon. Thank you.
So, thank you, More madam. Now, again, good morning, all of you.
Good morning, sir.
We are going towards second session of this second day, and it gives me great pleasure to welcome our second speaker, Dr. Gulab C. Maika from MQR Pharmaceutical Industry. Up till now, we have heard the lectures from academicians and researchers, but now at present, we are going to hear the industrial person who has huge experience of industry, that is work in pharmaceutical industry. About 50 years they work as director in MQR Pharmaceutical. So, sir had completed their graduation from Utkal, Orissa, India in 1985. Sir qualified CSR NET exam in 1988. Sir has completed their PhD degree from University of Delhi in 1992, and Sir has completed postdoctoral work in Indian Institute of Technology, Kanpur, India in 1994. Sir has industrial experience. Sir worked as a research associate from 1990 to about 1995 in Ranbaxy Laboratories, Dabur Research Foundation, Ghaziabad, Uttar Pradesh. Also, Sir worked as a research associate in Indian Institute of Technology, Kanpur. Sir worked from 1995 to about 2007 as an associate director in Dabur Research Foundation, Ghaziabad, Uttar Pradesh. Sir also worked as a director in MQR Pharmaceutical, Pimpri, Pune from 2007 to 2018. At present, Sir are the founder director of GPC Life Science Private Limited, Pune. Sir has published about 22 research papers in the reputed journals. Sir also published about 20 patents, and Sir has experience about work on anti-cancer drug, anti-HIV, and chiral molecules. Sir also have the experience about the scale-up and about method validation of the commercial drugs here. So with this short introduction, I request Dr. Mike Sir to deliver their lecture on drug substance specification and impurity profiling by using modern analytic techniques. So on behalf of our Professor of Pushmo College and all participants, I welcome Dr. Golak Maikap, and I request Maikap Sir to give a talk. Thank you.
Thank you. Thank you, Professor Rashale, for your kind words. So first of all, before I go into my presentation, so I would like to thank the organizers and especially Dr. Rashale for inviting me to give this presentation. So may I request Dr. Rashale to share my presentation? Am I?
Bange sir.
Hello.
Yes, sir. You are perfectly audible.
Okay. Thank you. So please start my presentation. Yes. Okay, that's fine. Thank you. So as Dr. Rasale mentioned, so today the topic of my presentation is drug substance, which is also known as API. The drug substance specification, or you can say API specification, the impurity profiling, application of modern analytical techniques. So when I say drug substance, so generally, so when you go to the market to buy a medicine, so basically that in our language we call it drug product. Suppose you buy a tablet, suspension, ointment, cream, whatever it is, because that is the formulated one. So that formulation contains some active substance which is responsible for the therapeutic activity. So that active substance is called as drug substance, or that is also known as active pharmaceutical ingredient. So that API, I will shortly call it as API. So whatever API is, manufactured by the companies, so those APIs are manufactured by a stringent process, which is, involved of chemical reactions and many more steps. So there will be sometimes 10, 15, 20 steps, starting from 2, 3, 4, 5. Okay. So each step is a chemical reaction. So this is, I will show, this is a pharma company at a glance. So, every process development for an API, because I will be more, concentration, give more concentration on API manufacturing rather than formulation. So how it starts? So every API manufacturing company has an R&D where the process development for the API or the drug substance starts. So R&D, then there is a technology transfer group, and then finally the process goes to manufacturing. It involves a lot of chemistry, lot of documentation, everything. And the research and development department or the group that is supported by QA. So there is no need to go further elaboration of QA. That is quality assurance, RA, regulatory affairs, EHS, very well, environment, health, safety. Then there is purchase department, there is analytical research department, ARD, and there is IPD, that is called intellectual property department. And then when it goes to manufacturing, so manufacturing is also supported by different groups, and in between there are groups like project management and microbiology. Because whatever drug substance we make, so that goes for human, that is used for human being. So even the stringent microbial specification is also there. Go to the next one. So this is the slide I am showing. So this is briefly I am showing what are the different steps involved in a process development. The process development of a particular API, for example, you say paracetamol, the simplest one I can take. So when I am given a project to manufacture paracetamol, so my job is to go and search the literature. What are the methods available? What are the manufacturing processes for the API paracetamol is available? So it starts with the literature search. So after literature search, I will find out a route which is green, which is commercially viable, which is scalable. Then that route, particularly that, I will start the lab experiments. Then process optimization and validation. Then scale up. Then I will finalize the specification. So there comes the analytical part. So for every chemistry, every step reaction, so I have to check the progress of the reaction, the starting material, the specification for the starting material, the specification for the intermediates, and also the specification of the final product. And after making the final product, I will put that drug substance for stability testing. That will be put under different conditions like accelerated conditions, long-term conditions, so that drug substance will be tested for its stability. Then it goes to the formulation department for formulation. Next one. Next slide, please. So these are the ICS guidelines, as I said, because whatever drug substance is manufactured, that goes for formulation, and that formulation is made for human use. So this ICH, that is the International Conference on Harmonization. So that ICH, so they have different guidelines, they have different chapters, and these chapters that control different steps of process development. That is Q1A to Q1F. Q1 is the chapter name, A to F, they are subsections. So that deals the stability. Then Q2 for analytical method validation. Q3, A to D, that is the impurities. What are the impurities present in a drug substance? How those impurities are controlled and how those impurities are monitored. Okay, so I will come to that later on. Then Q4 to Q4B, so that is about the pharmacopoeias. I think the audience knows what is the pharmacopoeia like. So whenever you buy a medicine from a druggist, so you'll find it will be written paracetamol IP, paracetamol USP, paracetamol BP, or cetirizine IP, cetirizine USP. So that USP, BP, EP, whatever it is, that stands for the pharmacopoeia. USP is United States Pharmacopoeia, BP is British Pharmacopoeia, IP is Indian Pharmacopoeia. So what is that pharmacopoeia actually? That pharmacopoeia gives the specification of that particular drug substance and the drug product. For example, paracetamol, how does it look like? What is the impurity profiling? Whether it is a solid or liquid, what is the melting point? What is the molecular formula? What is the molecular weight? So everything you will find in that pharmacopoeia. So that particular that paper, that part is called a monograph. That is called a monograph. So if you go and find out that monograph, so that will describe the different characteristics of that particular drug substance. Then Q7, that is says the GMP, you must have heard the name of GMP, that is good manufacturing practice. Then Q8 is pharmaceutical development, then quality risk management. So then development and manufacturing of drug substance, that is Q11. So this Q11 and Q3 and Q1 stability. So this is directly involved with the process development. Then Q12 is life cycle management. Go to the next one, please. Next one. Okay. So now coming directly to specification. As I said, so whenever you are making anything, so it has to have a specification. So that specification means that specification decides what is the quality of the substance you are making. What is the quality? What are the quality parameters? So the specification will contain different quality parameters. So a specification, so there will be a list of tests. Starting with the molecular formula, that will be there. That is the common parameters. Then there will be appearance. What is the appearance of the drug substance? Whether this is a solid or liquid. What is the appearance? And so this specification will contain a number of tests, and also it will give the limits. So what is the parameter? What is the range? What is the range of that particular test? For example, suppose you say purity. What is the purity? What is the limit? What is the limit? That this is 99%, 99.5%. Then there will be a reference how to test that parameter. So there will be tests. Then appropriate acceptance criteria will be there. Suppose you say moisture content. What should be the moisture content? Not more than suppose 0.5%. So when I say not more than 0.5%, that not more than 0.5% is the acceptance criteria. Because suppose you have made the substance having moisture content 0.6% or 0.55% or 0.7%, that means that will fail. That material will fail. So that is not complying to the acceptance criteria, which is 0.5%. Suppose you say LOD, loss on drying, or suppose you say residue on ignition. So these are very stringent parameters, even the impurity profiling. So when I say specification, the specification, then there will be the acceptance criteria at the middle. Then at the right hand side, there will be the reference to that test. How that test is performed. Suppose I say Fisher, suppose you have to determine the moisture content. How to determine the moisture content? What type of instrument has to be used? What type of reagents have to be used? So it consists of three parts. One is list of tests, then acceptance criteria, then okay. Then go to the next one. So go to the next one. So already covered this one. Next one, please. So why specification is important? Specification controls the performance of the drug substance, because the drug substance, that goes for the formulation. So if a drug substance is of inferior standard, then by using a drug substance which is of inferior standard, inferior in quality, you can't make a good drug product, good formulation. So the drug substance which you are making, that is the active part in a formulation, so that has to be specification compliant. So every specification, for every drug substance there is a specification, either it is in USP or IP or AP. Otherwise, if this drug substance I am making, it's a new one, suppose this is not covered in any of the pharmacopoeia, then I have to formulate my own specification, that is called in-house specification, but as per the ICS guidelines. Because ICS guidelines says what should be the metal content, what should be the impurity profile, what should be the known impurity, what should be the unknown impurity. So all these, every day is written in different guidelines are there. So I can make my own specification, but that has to be within the framework of the ICH or USP, EP, BP. So basically, our aim is to provide a quality product for the population. Go to the next one. So references, these are the, I have already covered. So QA, ICH, Q6A, that deals with the specification. Q3A to D, that is impurities. Then different pharmacopoeias are there, like USP, PP, EP, JP. JP stands for Japanese Pharmacopoeia. Similarly, if you go to any country, like in India, we have DCGA. Like in US, there is US FDA. That is called United States. Okay. So US FDA, Food and Drug Administration. So similarly, in British, they have a regulatory system. In Australia, they have TGA. In like in Russia, they have different regulatory system in there. So suppose I make a drug substance and I want to sell that product in the United States, because United States is the biggest market for pharmaceuticals. So most of the Indian companies, they actually compete to sell their product in US, either the drug substance or the formulated one. So if I want to market my product in the United States, then I have to follow US FDA guidelines. So that is the most stringent one in the world, you can say, US FDA. You must have heard many companies, they fail in audit. Because what is the process? Just like I'll tell you in just 2-3 minutes. So once you make your drug substance, you have to prepare a dossier. That dossier is known as drug master file. So once the formulation is completed, so that formulation department, they will file, that is called ANDA. So I am a manufacturing guy. I have made the drug substance. So I will file drug master file. Then my partner who has done the formulation, so they will file the ANDA, that is called abbreviated new drug application. Because I am talking about the generic products. Okay, these are the generics, but the patent is not there. Okay, so for generics we file generally ANDA. So if it is a new drug, new product, then you have to file ANDA, new drug application. And I am filing, abbreviated one, because some data is already, the clinical data, everything is available for that product. So I have to just prove the equivalence. Okay, so once I file my DMF as well as ANDA to US FDA, then that goes, under, rigorous, investigation. What is the impurity profile I have given? Whether I have followed the guidelines. So if they are not satisfied, so they will send some queries, like on NAC audit. So after audit, maybe sometimes you will receive some queries. So similarly, we also receive some queries. Then we have to address those queries. And once they are satisfied, so then they will give approval to the ANDA. And when ANDA is approved, automatically the drug master file gets approved. So after that, they will come for audit of your plant. And if they find everything, okay, so then they will say, yes, you can sell that product in US. And if they find any discrepancy, so they will send, discrepancy letters like, alert letters. Then so if they are very powerful, they can also tell you, you can't sell the product in the US. Warning letters, alert letters, you must have heard, Sun Pharma, MCure. So every company, Glenmark, every company, they are facing issues. Nowadays, people, now companies are learning the things. Okay, because earlier, the companies were not that experienced how to face the auditors first thing, and how to write the DMF and how to write the ANDA. So now everything is normal. Go to the next one. Okay, so this is the specification, So there will be description, because suppose you have made a solid product, solid drug substance. So whether this is white in color, off-white in color, or bluish in color, or greenish in color. So that is the appearance. Then identification, as per the SH guidelines. So these are the identification tests, like by IR, HPLC, UV, LC-mass, TLC. And you have to prove by at least 2 methods. Okay? By IR HPLC or IR UV, so you have to pick up 2 different methods. So you have to identify your product. Yes, my product, whatever that I have made, suppose cetirizine. So I have made cetirizine, so I can prove this product is okay by IR and also by comparing the retention time with HPLC. And suppose, most of the time you will find your drug substance is in a hydrochloride form, in a sulfate form. For example, levocetirizine or cetirizine, that comes as cetirizine hydrochloride or cetirizine dihydrochloride. So if it is dihydrochloride, the chloride ions will be there. So that is also qualitatively, you can say yes by doing synchronated test. If you find a qualified precipitate, you can say yes. So chloride ion is present. Go to the next one. So there are other parameters like assay, then counter ion content, water content, loss on drying. I think these are very much clear to you. And the assay, so assay is actual content. You know, when I say purity, when I say the purity of the drug substance, purity of a particular substance, it can be 99%, but the assay can only be 70%. Because suppose your substance contains a lot of water, lot of organic solvents, but the purity is not going to hamper because you are checking purity by HPLC. Okay, the purity can be 99%, but if you check assay, because assay is more quantitative one, there is a quantitative one. So assay gives the actual content, because that is very important. Because when you are buying a medicine, suppose 50 mg tablet, so 50 mg tablet suppose contains 5 mg of the drug substance. Because always the weight of the tablet is more than the weight of the active substance present. Because the formulated product contains a lot of excipients, lot of other substances, coloring matter, so lot of other substances. So finally, you will find the formulated product, the weight of the formulated product is more than the active substance. Like in paracetamol, 500 mg is there, or 650 mg is there. But if you take the weight of the tablet, that will be always more than 500 mg, more than 650 mg, because it contains a lot of excipients. Okay. So the actual content, that is called the assay. The counter ion content, suppose I say this is monohydrochloride. For example, suppose I say levocetirizine is a dihydrochloride. So I have to calculate, if it is a dihydrochloride, then how much chloride should be there by a differential method. So if it will meet that criteria, then I can say yes, this is dihydrochloride. Or suppose maybe sometimes if you have not adjusted pH properly, it can be a monohydrochloride also. So if you have made a monohydrochloride instead of dihydrochloride, so you can make a good product by using a monohydrochloride, because the solubility profile, the bio dissolution, everything will be different. So then your water content, that also plays a very critical role. Then loss on drying, pH, melting point, and residue on ignition. The residue on ignition, that shows the inorganic content, the metal impurities present in the drug substance. Okay, so that parameter is very, very stringent. Suppose it's a residue on ignition is not more than 0.1%. And suppose metal content, nowadays you are checking metal content by AS, ICPL, ICPOES. But these are parameters are getting very much stringent day by day. Go to the next one. Okay, so this is further and continuing. So these are the most modern one, like the polymorphism, the particle size distribution. So when I say polymorphism, because you will find many products which are available in the market. So that will be, what is polymorphism? The definition is very simple. Different crystalline forms of a particular substance. So suppose I say cetirizine. Suppose I say zoldronic acid. So zoldronic acid, that is a bisphosphonate which is used for arthritis. So that can exist in different polymorphic forms. But what happens? So for to get the right product, so I have to synthesize only the desired polymorph. And the polymorphs are numbered as either 1, 2, 3, 4 or A, B, C, D or alpha, beta, gamma, delta, like that. So I have to synthesize, I have to manufacture only the desired polymorph, so that you will get a better product in the market. You can put a better product in the market. Because the polymer is directly responsible for the solubility of the drug substance. Because when you are taking a medicine, so whatever the drug substance present in the formulated product, so that has to be soluble. Then only it will be bioavailable to the system. So the polymorphism also plays a major role in drug formulation and manufacturing of the particular drug substance. Then what is particle size distribution. Like always I give example, So always I give example when I take for students class, so when I say, maida, atta, suji, okay, then the higher, different particle size are there, okay, dalia and gehu. So what is the difference? When you say maida, atta, suji, dalia or wheat, So the difference is only the particle size. So the maida is more fine and atta, that is, slightly thicker. So similarly, when we make a drug substance, we give the drug substance to the formulation department. So they tell us, they give instruction. Yes, I want the drug substance of this much particle size. So sometimes they have a 3-tier specification, D10, D50, D90 like that. So if the particle, for example, why the particle size is important? Maida, the difference between maida and atta, right? Similarly, in my case, when I am making a drug substance, if the drug substance is very, very fine, then it will be more soluble. When the drug substance is more crystalline or slightly thicker, or the particle size is slightly bigger, then the solubility will be low. So that what should be the particle size, that is defined by the formulation department. When always we have to take, when we make the product, make the drug substance, we have to comply stringently to the particle size mentioned by the formulation department. Because if the particle size will be smaller and smaller, the material will be more fluffy. So when you go and buy a capsule, the capsule has a limited size. The capsule has a limited size. Suppose it's a 100 mg capsule. So I can put only 100 mg of the drug substance in the capsule. But if I make a drug substance which is more fluffy, so it is very difficult to pack 100 mg in that capsule. Okay, this is one thing. And secondly, the bioavailability also changes along with the particle size, and the solubility of the drug substance also changes. So because we are following SOPs, we are following different guidelines. Suppose I make cetrizine of different particle size, I take 10 batches, and every batch has a different particle size. Okay, for me as a manufacturing guy who is making drug substance, that is okay. But when I provide that drug substance to the formulation person, the formulation, suppose they have to dissolve that drug substance in water or in some solvent. So that solubility will differ, time-wise. So that means their process will be deviated. Okay. So I have to stick to the particle size which is given by the formulation department. Then the impurities, there are different types of impurities, organic, inorganic, degradation products, byproducts, side products, like that. Then one more important thing is chiral impurity. Because everybody knows, and I think you know very well, when you go and buy a product, esomeprazole, you must have bought, esomeprazole. Esomeprazole. So what is esomeprazole? You buy levocetrizine. What is levocetrizine? You buy Esomex, S-amlodipine. So these are the chiral drugs. So now that is the craze. We are making many chiral drugs. MQ, they started, first S-amlodipine. So now they have many products in the market, like dexketoprofen. So what is dexketoprofen? Ketoprofen is already there in the market. And dextroisomer of the ketoprofen, because that is the active form. So like cetrizine. So cetrizine is racemic. So cetrizine contains 50% R and 50% S. So maybe only one of the forms, when the drug substance is racemic, so in most of the cases, you will find only one of the enantiomer is bioactive. If one of the enantiomer is bioactive, then why to take the other one? 50% of the other one. Suppose I am taking 500 mg tablet, only 250 mg is bioactive, then why to take the other 250 mg inside my body? So that will put a lot of metabolic load on the liver. So that's why nowadays we are coming with chiral drugs like your cetrizine and levocetrizine. Cetrizine is highly sedative, but levocetrizine is not to that extent. Okay, so because the other isomer, the dextrocetrizine, so that has the sedative properties. The better case of thalidomide, that is responsible for a lot of teratogenic cases, because only one enantiomer is responsible for that. So if you can specifically synthesize the other enantiomer which is required, so then you can develop a good product. Then my point is, what is the presence, what is the amount of the other enantiomer should be allowed in the desired enantiomer. So that means you get specification. You can say, suppose levocetrizine, the dextro enantiomer should not be more than 1% or be more than 0.2%. When I make a drug, I have to follow this. Okay, this is called the chiral impurity. Then you know what is residual solvents. You are making the drug by following different processes, different steps. So every step you are using some organic solvents. So organic solvents are classified into class 1 to class 3. The class 1 solvents are highly toxic, like the benzene, carbon tetrachloride, 1-dichloroethane. So these solvents should not be used in a process. Basically, our aim is to use class 3 solvent like ethyl acetate, acetate. So these solvents are very friendly. Not friendly means the toxicity is comparatively less in compared to class 2 because they are highly toxic. Go to the next one. Next one, please. Okay. So then finally comes the microbial lipids. Because when I am making a drug substance, either that will be used in the formulation as a tablet or capsule or injectable. So tablet, capsule, you are taking oral. But an injection goes directly to the blood. So I have to be very, very careful, the presence of endotoxic and also the microbial proteins in the drug substance I am making. So go to the next one. So these are the limits for microbial contents. This is the limit I am giving for solid orals. The formulation is divided into different solid orals, injectables, live products. Some of the products are lyophilized because you go to the market, you will get some product. There are available 2 packs. So one will be a solid powder form, and then there will be one water. So when doctor gives the injection, so they will clear both the packs. So they add the solid into the liquid. They will make a clear solution. That solution should be very clear. Okay, then they will inject that. So that powder is a lyophilized one. So lyophilization is a process. And that water which is present, which is used to make that final injectable. So that is not ordinary water. That is called WFI. So that is water for injection. WFI. And that is the purest form of water, which is particle free, which is microbial free, which is there is no TDS. So that is the most purified water which is available. We can make. Okay, and that water also has a stringent specification. And every drop of water, every water which comes to our plant for manufacturing, so that is also on daily basis, that is also monitored for microbial content, for TDS, for other elemental impurities. So this slide shows the impurity profiling. The impurity profiling means suppose you have made a drug substance or drug product. I mean, this is particularly for API or drug substance. So what should be the impurity profile? So if your daily dose is less than 2 grams per day, then the reporting threshold, one is identification threshold and one is qualification threshold. So if your daily dose is less than 2 grams per day, so then the reporting threshold is 0.25. So if you are getting any impurity of HPLC or GC, which is more than 0.05, that has to be reported in your specification. If it is 0.03, then you can ignore. If the daily dose is less than equal to 2 grams per day. And identification threshold means, suppose you are getting one impurity which is 0.1%, 0.10%. That means that impurity has to be isolated, characterized. Okay, that impurity has to be characterized. You have to prove what is that impurity, what is the effect of that impurity. Whatever drug substance we make for drug product, that is very, very pure. That is very, very pure. Nowadays, we follow all ICD guidelines. There are different SOPs in place. You can't degrade yourself. Okay, so there, if the impurity is 0.15%, then that has to be qualified. Qualified means the method has to be validated. Then that impurity has to be quantified. How much impurity is actually present? Okay, and if the daily dose is greater than 2 grams, then the impurity profile becomes more stringent. You see 0.03, which was 0.05, that is 0.03, 0.1 is 0.05, and qualification threshold is 0.05%. Go to the next one. So quickly, okay. So how much time I have? Can somebody tell me how much time is remaining? Hello? Am I audible?
Yes, sir.
So how much time is remaining? Because another 15-20 minutes?
Yes, sir. Unmute, sir.
So, okay. So just 5 minutes remaining, just hello. Your type of impurities. Hello. Organic impurities there. Those impurities come under inorganic impurities. The residual solvent I told you. So in every step we are using some solvent like acetone, methanol, ethyl acetate, IPA, toluene, like these solvents. So what are the solvent? What are the residual solvent present in my drug substance? So that has to be changed by EC. Okay, and every residual solvent has a limit. And that limit is decided based on the toxicity studies. As per the ICH guidelines, as I told you, solvents are classified into class 1, class 2, class 3. So if it is class 3, then it is not more than 5,000 ppm. If it is class 2, the limit is very, very stringent, and that varies from solvent to solvent. If it is acetonitrile, the limit will be different, toluene limit will be different. And class 1 solvent, as I told you, the class 1 solvents are highly discouraged. That is not used nowadays in the process. And if it is used in a process, then you have to validate your method to prove that the class 1 solvents are absent in final drug substance. And then other impurities. The other impurities are very, very important. Now go to the next one. Next slide, please. So these are the organic impurities. So I have to go to the next one. Go to the next one, please. Okay. So inorganic impurities, I told you, organic impurities, I told you, because my concern, I give more emphasis on other impurities. So when I say other impurities, organic impurity, inorganic impurity, volatile residual, residual solvents, metallic impurities, they are very well known. When I say other impurities, excluding all these impurities, what are the other impurities present? Like, elemental impurities. Elemental impurities like your heavy metals, some of the metallic impurities are present. Because you are using some reagents in the synthetic process, like palladium, rhodium, lithium, sodium. So those residues will be present in the drug substance. Okay, and whatever present in the drug substance, that will go into the drug product. Right? Okay. So elemental impurities, then polymorphic impurities. I told you. I mean, suppose your desired polymer is a form, and you are making a drug substance which also contains certain amount of beta form. Okay, so that beta form has to be specified. How much beta form is there? Whether that is within the limit or not. Okay, these are the polymorphic impurities. Then there is one more impurity that is called extractable leachable. So extractable leachable, what is extractable leachable? Because suppose you are packing your drug product, injection. So injections are put inside glass bottle. And those glass bottles are very different from, the cold drinks bottles or your oil bottles. So those are glass. Those glass is highly pure, and those glasses are leaching proof. Okay, nothing will leach from the glass. Nothing silica, nothing elements. Nothing will come into the product which is stored in the glass. So just to check, sometimes you will find some precipitation. Okay, you will find some injectable, has some precipitation, has some particles. Then that undergoes, thorough investigation. Where from that particle has come? Whether that is released out from the glass, or some drug product which is getting precipitated, or the drug product has been degraded, it has not been stored properly, either it has undergone thermal degradation, or it is not packed, or when it was packed, some oxygen was inside. Okay, and oxygen can cause oxidative degradation, or the pH of the drug product is not suitable for the particular type of glass, or the stopper which is put in the glass vial. that is not suitable, they are not compatible with each other. So there has some interaction taken place and some precipitation has taken place. You are getting some particles. Okay, so this is not extractable leachable. Whether something is being leached out or something is extracted, something is extracted from the stopper or something is extracted from the glass vial. Okay, so I mean this is a very stringent process in formulation. What they do, suppose you find in the drug product only half of the glass vial is filled, 50%. So in order to study this leachable study and leachable study or extractable study, because the stopper which is used, that is also Teflon or rubber sometimes. So they will keep the sample inverted, so that most of the drug product will be in close proximity or in contact with the stopper. So that study will be done. So then they will make if something has been leached into your drug product. Already I have told you what is enantiomeric impurities. So we are making leukocytin. So what is the amount of dextro? What is the amount of dextro? Suppose I am making esomeprazole, or suppose I am making esomeprazole. Then what is the amount of R-omeprazole present there? Okay, so that has to be defined with particular limit, and there has to have a particular analytical method. Because when I am saying everything, so it is related to the analytical method. Because the analytical method has to be very stringent, because the impurity profile I said at 0.03%. If the daily dose is greater than 2 gram, then it should be 0.03%. Then the method has to be very, very specified. The method has to be very robust. The method has to be validated. To detect impurity up to that level, 0.03%. The instrument has to be very, very sensitive. Okay. Suppose sometimes your TB detector is not good enough, then you have to go for mass detector. Okay, we'll come to that. Then finally, I have put in bold here genotoxic impurities, because most of the drug substances we make, they may contain some potential, what we call it PGI, the potential genotoxic impurities. Go to the next one, please. So genotoxic impurities, I can divide those genotoxic impurities under mutagenic impurities, carcinogenic impurities, and clastogenic impurities. So all these are that the dividing line is very thin. So they are all mutagenic. If I say, so they are all mutagenic, they can produce mutation in your gene. Okay, because they interact with your DNA, they interact with your nucleobase. So because nucleobase containing nitrogen, that is nucleophilic in nature. Okay, so any drug substance containing some impurities which is having a limiting group, for example sulfonates, for example your halides, so they can act as a genotoxic impurity. Okay, so mutagenic, which can produce mutation in your gene, carcinogenic, which can cause cancer, and clastogenic, which clips the structure of your DNA. Okay, go to the next one. So this is, I have shown, this is your DNA and how the base pairing is there, adenine, thymine, guanine, cytosine, all those things are there. Go to the next one. Go to the previous one. Go to the previous one. So you see these base pairs are basically by 2 hydrogen bonds or 3 hydrogen bonds. So if you can break that hydrogen bond or some intercalation is there, so all these nucleobases, whether this is purine base or pyrimidine base. Pyrimidine means single one ring, and the purine is fused ring system. Like your adenine and guanine. Adenine and guanine, they are purine base, and thymine, cytosine, and all this. So they are pyrimidine base. So I mean they are having nitrogen. So if the nitrogen, which is responsible for hydrogen bonding, now that attacks a substance which contains a lipid group, that means this hydrogen bond is cleaved. First thing. Secondly, some additional substance is getting attached to your DNA. Okay, and if that additional substance is that can cause mutation in your gene. Go to the next one. So nowadays, what is happening? So when you are making a reaction, we are doing a reaction, we are making a drug substance, we are checking what are the potential genotoxic impurities present. If there are genotoxic impurities, then we try to purify the drug substance. Okay, and if it is not possible to purify, then we have to define. So there are guidelines. That guideline says how much genotoxic impurities are allowed. How much is allowed. So then that has to be checked by a very stringent analytical method like SMAS or GMAS. So these are some of the genotoxic, they are probable genotoxic impurities because they are all electrophilic in nature. You know, like your mesylate, because in suppose in reaction you are using some methyl sulfide chloride, and suppose there is some alcohol in the solvent, so that alcohol can react with methyl sulfide and give you alkyl methyl sulfonate. Similarly, mesylate, alkyl halides, you are using methanol in a reaction, and you are using a PCl5, you are using hydrochloride, so there will be generation of some halide genotoxic impurities. Like similarly, you are using hydrazines. You are using some, polyaromatic hydrocarbons. So they are all genotoxic impurities. Okay, go to the next one. Next one, please. Okay, so this is to determine. Suppose you find the potential genotoxic impurities are there. So nowadays, different softwares are available like Derek, Multicase, Topcat. Okay, these are different softwares. They will give you prediction. If you give the structure, in the system, so that will tell you, what is the probability of that particular substance to be a genotoxic one. And if the probability is very high, if the score is more than 60%, 70% or 80%, then finally you have to isolate that substance. Then you have to go for a AM test. That is the ultimate one. So AM test, that is the bacterial assay test. That will tell you whether that substance is genotoxic or not. Go to the next one. Okay, here I have shown this is what I will paper cutting. So the pharmaceuticals, they have, sometimes what happens, the some products I have already launched in the market, but then I find that that product is, some genotoxic impurity. That product contains some extra impurity which was not detected earlier. Then I have to withdraw or recall that product from the market. So there are different types of recall, like type 1, type 2, type 3 recall. Okay. So before it goes to human beings, so I can recall that product. Or even if it goes to few population, then I can recall that product. Okay. Because sometimes because the stability testing that is undergoing. Okay. And I find suddenly some impurity which was not earlier present has appeared. Then I have to recall that product. Go to the next one. Like one genotoxic impurity, just about the name of NDMA. Because sometimes what happened, last 2-3 years, because most of the certain drugs like Balsartan, Telmisartan, and also ranitidine and all those drugs, they were found to contain one genotoxic impurity. Okay, so that is called nitrosodimethylamine or nitroso, because it is nitroso compounds are genotoxic. Nitrosodimethylamine. So many products were, withdrawn from the market. And US FDA, they started, these impurities were not earlier detected. So then US FDA started, auditing the plants where this impurities has come. Because maybe my analytical method was not sufficient good enough to detect this impurity. That's what I'm saying. We give a lot of emphasis on the analytical method development. So analytical method should be capable to detect all the impurities. So the next one is. So then, polymorphism, already, they are very simple terms. So any drug substance, solid drug substance, that has a, potential to exist in different polymorphic forms. And I have to take only one polymorph, which gives better dissolution profile, better bioavailability. Okay. And sometimes these polymorphic forms, they interchange under heating, under light, under photochemical air. So that's why. So when you buy a drug product from the market, you have to follow stringently what is the storage condition mentioned. Whether the drug product has to be stored away from light or away from heat, away from moisture, because all these instructions are given by doing stability study, by doing duration studies. These are very, 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 very important instructions. Okay. So you have to, when just I'll give you one example. So suppose you are getting, you have got a bottle which contains 30 tablets, and your doctor says you have to take one tablet daily. So that means how many times you will open that bottle? 30 times. Okay. So we in the formulation we do one study. we check the stability by opening the bottle 30 times. Product, be very careful. So when you buy a drug product, you have to follow the instruction. And when you take out one tablet, then immediately close the bottle. Okay, and keep it in a desired storage condition. Okay, like in different forms. And in polymorphs, you find there are hydrates, there are solvents, and there is also amorphous. So these are different crystalline polymorphs. Solvents, hydrates means suppose you will find one drug substance is monohydrate, dihydrate. Just one inorganic example. For example, copper sulfate. For example, sulfate. Okay, I am talking about drug substance which is quite different, but just to make you more familiar. So when you zinc sulfate is 7 hydrate, or copper sulfate is pentahydrate. Similarly, there are drug substances which are monohydrate, trihydrate, dihydrate, like that. Suppose I have to manufacture a drug substance which is dihydrate, then always it should contain 2 water molecules. So each molecule of the drug substance should be hydrogen bonded or by anyhow. So it should contain only 2 water molecules. If there will be more water molecule, then it is not the drug substance which I want. Okay, so this water content has to be manipulated. And this polymorph, it is hydrate, because when hydrate or solvent is there. For example, acetonitrile. So acetonitrile can also be bonded to some of the drug substance molecules. So in that case, when it is a solvent, we call it a solvent. When it is water, we call it hydrate. And these hydrate or solvents, they are called pseudopolymorphs. They are not actual polymorphs. When the actual polymorph we say, when the crystalline structure is different. And when this pseudopolymorph means these water molecules are trapped inside different crystals. Okay? So the crystal structure is different because of the presence of the water molecules, solvent molecules. So these are called pseudopolymorphs. And amorphous, what is amorphous? It has no clear cut or well-defined crystallinity. So that is called amorphous. So in amorphous substance, you won't get a sharp melting point. Okay, it will get melting point in the range. So it will, start from suppose 80, it will go up 120. But when it is a crystalline form, that gives a sharp melting point. Go to the next one. Next one, please. Okay, so these are, when I say these polymorphs can be detected by different methods like IR, Raman, DSC, PGA, DTA, then solid state NMR, then mostly what we use in pharmaceutical area, because mostly we make powders, so we go for powder X-ray. So that is called PXRD. We do X-ray, that is called powder X-ray. So that will give you whether you have made the right polymer or not, and even also by what is the contamination of the other polymer or other polymorphs. Sometimes drug substances are available having more than 10 different polymorphs, 15 polymorphs, 5 polymorphs. So it is very challenging to make the desired polymorph in kilogram quantities, in tonnage quantities. Maybe it's very easy to make 1 gram, 2 gram in the laboratory. But when I develop a process in the laboratory, my aim is to make a process which is very robust, which is scalable, so that eventually I can make the drug substance at a desired level. If the drug substance is in kilogram quantities, then my process should be accordingly, very, very robust, so that when my process goes to the plant to, for manufacturing of kilogram quantities, 10 kilogram quantities, or tonnage quantities, there should not be any issue with the process. I should get the same impurity profile. I should get the same yield. I mean to say I should get the same quality of product. So it's not that easy. It's not that easy. So it involves, lot of hard work, lot of knowledge, and lot of experience. Okay, go to the next one. So these are the different, we compare, like I have given the example of zoldonic acid. This is zoldonic acid. So there are 2 phosphonates. There are 2 phosphonic acid groups. So this is the XRD, 2 theta value, the D spacing and relative intensity. So this is just I have given an example. So this is the X-ray diffractogram of a particular polymorph of zoldonic acid. So this is powder XRD, powder X-ray diffractogram. Go to the next one. Next one, please. Next slide, please. Okay, go to the next one. Go to the next. Okay, so this is as I told you, suppose one drug substance has different forms, polymers, form 1, form 2, form 3, form 4, form 5, and this is the FTIR pattern. So the FTIR you can also differentiate different polymers by doing FTIR, and also you can differentiate by doing solid state NMR and by doing DSE. I will go one by one. So there is slight differences are there in the FTIR pattern. Go to the next one. So you see the DSE, differential scanning calorimetry. So this is just trying to find out the melting point. Okay. So you see that I am given here the lipid substrate. This is a co-crystal form. Okay. So if I take the DSE of different forms, form 1 and form 2, their DSE pattern is different. The onset, peak. You know about DSE, right? So they will have different peaks, different melting point by DSE. Different polymers, they have different melting points. Okay, that melting point sometimes it is very very close. Maybe they differ by 1 degree or 2 degree, but you can differentiate by doing differential scanning calorimetry. So this red one is one form, that is form 2, and this black one, that is form 1. You see the onset, or you can say the peak for the black one is 178.4 and other one is 184.7. But here in this case you are very lucky. The difference is very, wide difference is there. But sometimes the difference is very big. Go to the next one. Okay, so this is the solid state NMR. Here I am shown a compound, indomethacid. So here you see the C13, the liquid NMR and solid NMR. So they are quite different. So this, by doing this solid state NMR, also you can find, here the polymers are named as alpha, gamma and amorphous. All their NMR patterns are different on the right hand side. But by doing a liquid NMR, you can hardly differentiate between alpha, gamma and amorphous. Okay, but here by doing solid state NMR, so you can easily identify which one is the alpha form and which one is the amorphous form. Go to the next one, please. So go to the next. This is a very simple slide. Go to the next one. This is showing the chirality. So enantiomers, you know what is enantiomer and what is diastereomer. Go to the next one. Okay, so this is by doing HPLC, sometimes we have to do chiral HPLC. Suppose I have manufactured, like this is the case of cetirizine. Suppose if you inject cetirizine in a chiral HPLC, when I say chiral HPLC means HPLC where you are using a column which is chiral. Okay, the column contains a chiral material. And suppose this cetirizine is 50% levocetirizine and 50% dextrocetirizine, because cetirizine, so there is a chiral center. Okay, so chiral center which is attached to 4 different groups. Any carbon attached to 4 different groups is chiral. Is asymmetric. Asymmetric is always, we can say this is racemic. It contains 50% R, 50% S. So you can try to separate those racemic into R and S. Again, R, S, they can be plus or minus. This is optical isomerism. Okay. So this cetirizine, if you take and inject cetirizine in a chiral HPLC, that reference is there. Chiral HPLC will get 50% of dextrocetirizine, which is coming at 23 minutes, and levocetirizine, which is coming at 27.64. So if you have made some levocetirizine, okay, you don't know how much dextro is present. So if you follow this system in HPLC, then you inject, then you can find out how much dextro is there in my cetirizine, levocetirizine, how much dextro is contaminated. So that can be found out. So this is called chiral HPLC. This is, okay. Now we are using nowadays very, very frequent in this chiral HPLC because more and more products are coming which are very chiral, chiral in nature. So this is a chiral system which is developed nowadays. So we have used, but what is the basic principle? Because when these enantiomers, they don't differ in their physical and chemical properties. So their physico-chemical properties are same, right? But once they go inside the chiral column, they are converted into diastereomers. With this chiral column, this chiral column contains some chiral packing materials. Okay, so this chiral packing material will interact with cetirizine R and cetirizine S. Okay, so making it converting into diastereomers. And once there is a diastereomer formation, then these physicochemical properties differ. Okay, so one will elute first and the other one elute after that. Okay, go to the next one. Okay, so similar, this is a similar kind of thing. Okay, cetirizine, in cetirizine, what happens? Cetirizine may contain, suppose you have made levocetirizine, it may contain some dextrocetirizine, so that can be detected by HPLC. And the impurities, it could be that the cetirizine or levocetirizine impurities are also chiral. Okay, because when the impurities are chiral, so the impurity will also give two different peaks in a chiral color. Right? So for impurity also, you know, the impurity, this is CH2COS, and cetirizine, you know, this is OCH2CH3. So the impurity, this is a hydrolyzed impurity. Okay, so the impurity can also give two different columns provided the impurity is chiral in nature. Okay, go to the next one. So this is GC. So like chiral HPLC, similarly chiral GC is there. For example, I have shown you know plus menthol minus menthol. So they can be distinguished by doing chiral GC. So that will give you, you know, minus, and that will differentiate. You will get two different peaks. You will get two different peaks for plus menthol one peak and for minus menthol one peak. Okay, go to the next one. Next one, please. Okay, so my point is, you see now this drug substance development or drug product development, that is always, you know, associated with this modern analytical techniques. For example, forget about those traditional impurities. You know, those traditional impurities, for example, metal content. Just I will give one example. This metal content, these heavy metals, which include, you know, 10 different elements like lead, bismuth, mercury, tin. Okay, so these 10 different elements, there was a qualitative test by USP. You know, it should not be more than 20 ppm, and that is a qualitative test. You make some solution, you observe the color. So that is not a quantified one. That is a semi-quantitative one, this color. And this color perception can vary from person to person. So now USP has changed. So this qualitative test is not allowed. So you have to quantify which element in what percentage is present, in what ppm. So that has to be determined by ICP OES. You know, you have to go, this metal has to be detected by either AS, atomic absorption spectroscopy, or the ICP OES or ICP AS. Okay, similarly the chiral impurities, these chiral columns have to be used. So one has to be acquainted with these chiral columns. And polymorphism, you have to use, you know, X-ray, powder X-ray, you have to use FTIR, you have to use solid state NMR, you have to use, you know, DSC. And one has to be very acquainted with all these impurities, all these, you know, modern analytical instruments. Because as you see, the drug substances are getting more and more complex nowadays. Nobody is making, you know, nobody is discovering paracetamol type drugs now. It's very, very, you know, there people are working. But nowadays you see remdesivir. You know, remdesivir is a very complex structure because I myself is making remdesivir intermediates. I know it involves, you know, lot of chemical reagents, lot of hazardous reagents, many Grignard reagents. So when I am using a Grignard reagent, which is magnesium, or I am using some copper-based, zinc-based reagent, so my drug substance finally may contain certain amount of zinc, certain amount of magnesium. So how to ensure that my drug substance is devoid of, or it does not contain, or it contains within the specified limit. So I have to go for all these instrumentation techniques. Okay, so go to the next slide, please. Okay, so this is, I have given another recall, because you know Sun Pharma has recalled one drug product because of the presence of some leachable impurities in the vial. They had recalled the drug. Okay, so I have to check what are the effect of leachables present, particularly for injectables. Okay, So nowadays, you see, it's very, very stringent. The impurity profile is very, very stringent because the basic purpose, the basic idea is whatever goes to human beings, that has to be the very, very pure one, provided in the parameter, within the framework, whatever the instruments available, the techniques available. Like I told you in the ranitidine or some certain drugs, some is dimethyl urea, nitroso compounds are found. But at that time, those compounds were not detected by the method. So particularly people who are working on the analytical side, their responsibility is very, very high. So they have to develop a very robust method. So that should be able to determine all the impurities present. Of course, in collaboration with the synthetic people, because the synthetic person who is working in the drug substance, they have a very good idea. So finally, you see, this, in order to make a good product, so this is teamwork. Synthetic chemist, analytical chemist, good formulator. Okay, so all basically good project guy, good microbiology guy. Okay, so it is basically a team effort. So if the team works properly, then we can develop a better drug substance. We can develop a better drug. Okay, for the human society. Go to the next one. Okay, so like as I told you, suppose for genotoxic impurities, so whatever analytical method you develop, that method has to be validated properly. And genotoxic impurity, the limit is so less, so it is very difficult to validate the method by GC or HPLC by using a UV visible detector or PDA detector. Or so we have to go for LC mass. We have to go for GC mass. We have to use mass as a detector. Okay, to develop the stringent method, to develop a detected method. Okay, I think this is the last one. Next. Thank you. So this is all what I I had thought to convey to the audience. So hopefully, so I have tried my best. So now it is for discussion. So any query, I will try my best to explain if there is time.
Thank you, Dr. Michael. Anybody have any question?
Sir,
you can ask that.
Yes, sir. So the one query is there. Sir, on slide number 25, you mentioned the SS NMR. So can you please explain what is the difference between the normal NMR which we are taking and this SS? Yes, sir. Just yes. Slide number 25.
Yes. Go to the next slide.
Yes, this one. Yes.
Next. Yes.
Yes.
Can you I think it cannot be magnified. Okay. This product I have never got myself. So I have from the okay, see this NMR. So this is the liquid NMR. Okay, so the substance or the compound has a potential to exist in different polymorphic forms like alpha, beta, and amorphous. Alpha, beta, and gamma, they are crystalline forms. Amorphous. Because by doing a dissolve, when you dissolve a solid in a liquid, so this polymorph does not exist. Okay, so you will get a similar pattern. Your liquid NMR. But if you are solid state NMR, because their crystalline nature is different. So you see in solid state NMR, so you can easily see the difference. If you can magnify, then you can find out what the difference is. But you can see, there easily see to the naked eye. So this solid state NMR actually differentiate between the different polymorphic forms. This is one of the techniques. This is not the only technique you have to use. You have to do solid state NMR and also.
Thank you, sir. Thank you.
Any more question?
Hello. Yes, sir.
Yes.
Am I audible?
Yes.
Sir, how we can change the chiral property of molecules? Which method or which technique we should have to apply?
You want to change chiral properties?
Yeah.
No.
So how to change it? I want to know that.
No, I am not getting your question actually, because what happens? Suppose, for example, I say thalidomide, okay, or you say cetirizine. Cetirizine is a racemic substance, Okay, it contains 50% one enantiomer and 50% other enantiomer. But suppose your aim is to make only levocetirizine, or only dextrocetirizine, or only esomeprazole. Then from the beginning, what happens? You have to do some asymmetric synthesis. This is one example. Asymmetrically, you should try. For example, you know thalidomide. Thalidomide, you see, one of the enantiomer is teratogenic in nature, and the other enantiomer is very safe. Suppose I want to make only the S enantiomer which is very safe, okay, or the desired enantiomer which is safest enantiomer, then I have to design a synthetic, synthetic scheme accordingly, so that I should get only that. I should end up with only that enantiomer. Literature methods are there, okay. And some other times, what happens? What the industries follow. Suppose, for example, let us take the case of ketoprofen. Okay, ketoprofen, what happens? In that case, we don't do chiral synthesis. We do chiral resolution. Ketoprofen, which is available in the market, that contains 50% R and 50% S. Okay? And S ketoprofen is the desired one. And S ketoprofen is always, suppose, S class. That you can't change. Okay? Suppose you want to say you want to make S without doing chiral synthesis. So how to achieve that one? You have to do a chiral resolution. You have to do a resolution. How? Because ketoprofen, suppose that has a COH group, okay, and it contains 50% R and 50% S. You take a chiral reagent, for example, some phenylethylamine. You take a chiral, alpha phenylmethylamine, which is chiral, and that is a base, okay. And this ketoprofen, which is 50% R and 50% S, this is an acid, okay. And if you are reacting with that acid with a base, so what happens? So this R acid will react with the base, and S acid will react with the base. So they will make different salts, okay. Because since I have taken this resolving agent, this amine, the chiral amine is a resolving agent. Suppose chiral amine I have chosen only the S. Suppose I have taken the S alpha phenyl methylamine. Now R will form a salt with S, so we will get RS salt. Now this S of ketoprofen will react with this S amine, so we will get SS salt. So now this RS salt and SS salt, they are physicochemically different. Their properties are different. Their solubility is different. So I can do, by crystallization method, I can isolate one form, which is a salt. Then, if I treat this salt with some acid, some mild acid, so I can generate only the one enantiomer, the desired one. Okay? And I am coming to another point that is also very interesting. So what happens? Suppose I want the S enantiomer. So what will do with the R1? The 50% of the R1 goes into first. Okay, so that is a loss for me. So what I do, I take that R1 means the R enantiomer, and treat with concentrated alkali. So when I treat, there is one hydrogen, hydrogen is there. So when I treat with alkali, so there will be racemization at that chiral center. So that R will again be converted into 50% R and 50% S. So again I can follow this resolution process. This is a tedious one, but this is the cheapest one which most of the industries follow. Clear?
Okay, sir, clear.
Okay. Thank you, sir. Now may I recall one of our participants. May I to propose vote of thanks to Dr. Michael.
Yes. Thank you, sir. Good afternoon, all. It gives me immense pleasure to deliver vote of thanks for the session in this FDP program. I am thankful to our respected speaker of this session, Dr. G. Maikap sir, ex-director NQF Pune, for giving valuable information on the drug substance specification and impurity profiling. Sir, you have explained different tests and analytical procedures with numerical limits and range, and also different types of impurities in the drug substances. especially the genotoxic impurity, in very informative manner. Sir, you have honored this session with your valuable talk. I am also thankful to Ramakrishna More College and organizing committee of the program for providing me opportunity to participate and propose a vote of thanks. Thank you all.
Thank you, Megha.
Thank you, sir.
Okay. So this is the end of your second session. So there is lunch break for up to 1 o'clock. Join session at before 1 o'clock. Don't be late. Yesterday we see that in last session so many are absent or so many leave earlier. So don't do that like that here. Join before 1 p.m. So thank you very much. This is now lunch break.
Okay, thank you.
Okay,
I'm
thank you.
leaving. Okay, thank you.
सुरू करायचं. एक वाजले. आवाज देऊ. गुड आफ्टरनून मॅडम.
Good afternoon, sir.
Okay, welcome to all the participants and speaker today's afternoon session speaker Jagtap Madam. I'm very happy to introduce afternoon session Jagtap Madam, working as a professor at Baburaji Golab College, Sangvi. Madam has completed MSc degree from Mumbai University, and he got gold medalist, and also he's a recipient of several merit scholarship from the Mumbai University and Department of Chemistry. Madam also passed NET exam, SET exam, and GATE exam, and also PhD completed from National Chemical Laboratory, Savitribai Phule Pune University in 2007. Madam also postdoctoral research as visiting scholar at Stanford University, California, USA, under the guidance of Professor Barry Trost from 2014 to 2015. Madam studied computer-aided drug design and quantitative structure activity relation in renowned institutes under well-known scientists. Madam completed postgraduate diploma in industrial program in pharmaceutical chemistry and production. Madam has 27 years teaching experience at Baburaji Golab College, Sangvi, and he has also worked as a PG principal in charge 3 years at PDA Arts Commerce Science College, Sambhajinagar, Chinchwad, Pune. Madam also recognized MPhil PhD guide, and he has pursuing 5 students PhD student under his guidance. Madam has 16 years teaching experience, research experience, and research area on catalysis, methodologies, and synthesis. Madam also presented national and international conference papers in various countries like USA, Australia, Germany, Singapore, Spain, etc. Also completed 2 minor research projects funded by UGC and DCUD, Savitribai Phule Pune University. Also published the highly impact factor, high journal impact factor journal. 30 research papers are published. Madam also published one book, Pharmaceutical, Medicinal and Natural Product Chemistry, co-authored with Professor P.S. Kalsi. This is the brief introduction, and welcome Jagtap Madam, and start the session.
Thank you, Gadwe sir. Thank you all the RMC team for inviting me. So I'll quickly start with the presentation now. Is the screen visible properly?
Yes.
Yes. It's not moving, right?
Hi.
Okay, I'll just see. Slides are moving? Gadwe sir?
Slide moving, but full screen nahi hai.
Yeah, full screen.
Slide show
मध्ये full screen मध्ये नाही येत आहे ते.
Presentation mode मध्ये नाही दिसत.
I'm doing it, but now is it okay?
Yes.
Okay, sir. Thank you so much for your introduction. And now I'll quickly start upon with the mass spectroscopy. So I hope people are ready to listen, because now it is post-lunch session. So hopefully you will enjoy this. I'll give the proper justice to this lecture. Okay, so let's start with this mass spectroscopy. It is the one what we have in our MSc syllabus generally, and what we teach to the students is in 2 sessions basically. Actually, organic people study it in a different way, analytical people study in another different way, and biology people study it in their own way. Okay, so here I have tried to club all 3 things. First, I'll talk about in general about the introduction, like what is mass spectroscopy and the basic principles and the history and all things. Then I'll just talk about the instrumentation, which is an analytical point of view is going to be important. So I have just collected all the information. We'll see some organic chemistry applications, how the mass spectroscopy can be used for the organic chemistry, and of course the biological molecule to study. Voice is not coming. Nadave sir, can you hear me?
Voice is audible.
Yes, come, madam.
Yes. Okay, thank you. So, and for having the application in biology, it is going to be itself is a like 2 lectures topic. So I've just taken the glimpses of that and talked in the advanced mass spectroscopy method. So here in the introduction, what we will see first, what is mass spectroscopy. So mass spectroscopy is an instrumental technique in which the sample is converted to rapidly moving positive ions by electron bombardment, and charged particles are separated according to their masses. So here we actually have the masses, the particles, charged particles, especially the positive charged particles which are coming out. We will see that in due course of our lecture. So these particles, which along with their charges, we are going to record it, and we are going to get the mass spectrum of this as a what we call as a mass spectrum, which is a plot of relative abundance against the ratio of mass to charge. Okay, so it is different than the other spectroscopies. We know that UV, IR, NMR, these are the spectroscopies. We actually call them as a, especially UV and IR, we call them as a true spectroscopies, because spectroscopy indicates it is a spectrized light, scope is to view. So we are going to view actually some molecules in presence of light, like is in UV and IR. Mass is not true spectroscopy because we don't use the light here directly to visualize the compound. We are actually going to fragment the molecules. We're going to break the molecule and study the smaller particles. But still, yes, mass spectroscopy, these spectrometers are the most powerful analytical tools in chemistry. Many people nowadays, along with the UV, IR, NMR, the mass spectrum, they analyze it to understand basically the structure of the organic compound. So here we use the mass spectra in 2 general ways. To prove the identity of 2 compounds, we'll see one example of this, and to establish the structure of new compound. I've taken one example for this also. I don't know whether I'll be able to take that, but let's see. Okay, so these 2 main important uses are there nowadays we do in the research. So mass spectrum is helps in establishing the structure. It can give you the exact molecular mass. So what information we get is a molecular mass, and which can give you the structure finally. So when you look back to the history of mass spectroscopy itself, J.J. Thomson, who is the inventor of electron in 1897, he invented the electron, and in 1906, just 9 years after that, he just gave a prototype to measure the mass to charge of electron, and for that he was awarded with the Nobel Prize also. Then after 6 years, he introduced the first mass spectrometer, which was originally called as parabola spectrograph. So that was constructed by him. And of course, then the later the newer versions came, where the Francis Aston, who put this concept into practice and designed to measure the mass of elements, and he was also awarded with the Nobel Prize. Then a very important analyzer came into a picture, that is time of flight. Then the Paul introduced quadrupole ion filters and invented the ion trap and was also awarded the Nobel Prize. And then in 68, we got a tandem mass spectrometer. All these things we are going to see in detail. But you will notice one thing is there are 3 Nobel winners, prize winners are there for the mass spectroscopy invention. So, of course, when you start with the mass spectroscopy, just let us understand the very, very basic of your mass number. What do you mean by mass number? So, which is also known as mass number or nuclear number, is nothing but the total number of protons and neutrons in an atomic nucleus. So, for carbon, there are 6 protons and 6 neutrons are there. So, the total mass number of carbon becomes 12. So, 12 for the carbon it is there, but for other elements, we have in fractions. Exact 12.0 is the atomic mass of the carbon, but others will be in fractions with relation to the carbon they are measured with. So how does mass spectrometer work? So here what we do is 3 things mainly. We do the ionization, that is what we do there is a create ions, then we separate these ions, which we use it by the analyzer, and then these are detected, so we have the detectors there. So 3 main components what we will be seeing in detail afterwards, but this is the main, the mass spectrometer works. Okay, so spectrum what we get out of each compound is unique, and therefore we also call mass spectroscopy as a chemical fingerprint. And that can be used for the characterization. So basic principle, as I said, first the organic molecules are bombarded with the electron, or there are many other ways also. We will see shortly all these methods. So they are ionized first. These ions then move rapidly with the accelerator, and then they have some kinetic energy with them, and then depending upon the charge they carry, they will be moving, and then these mass to charge ratio, these particles are actually detected, and these detected particles will give you some spectrum which we call as a mass spectrum. So this is what is, you can see the general instrumentation here. This portion, if you can see the mouse, this portion gives you the ionization, and then the acceleration is done in the next portion here, and then the detection will be done, and then the deflection will be there, and then finally they are detected. So each step we will see in short. So here the mass spectroscopy works in this way, that first they are ionized, then accelerated, then deflected, and they are detected to give the mass spectrum which will look like a bar chart or bar graph. Okay, so here we get a stick diagram where the abundance versus mass to charge ratio, these are plotted. So this is the basic theory, that is first the deflection will be there. Deflection is less for the heavy particle as compared to the light one, and you get a quick other reactions there. Okay, so as I said, there are 3 main stages which are there. The first one is going to be the ionization. So ionization is nothing but the atom which is ionized by knocking 1 or more electrons off from the that atom, and it will give you the positive charge, right? And then this will be bombarded with the stream of electrons to knock off 1 or more electrons, and these positively charged will be moving further. Okay, so generally they have a positive +1 charge, but there are evidences that there are +2 and +3 charges are also there. Then they are accelerated. The accelerated ions then will have the same kinetic energy, and where the positive ions are repelled away, and all the ions are then finally goes to the focus beam. So they are, whatever they are accelerated, they will be going to the focus beam. Then there is a deflection where a typical curvature is there into that part, and where the ions are then deflected by the magnetic field. So these are the heavy magnets which are on the sides here, and according to their masses, the lighter ones they are, the more they are deflected. the amount of deflection also depends on the number of positive charges on the ions. So how much is the positive charge? As I said, generally it is +1, but there can be more than that. So amount of deflection will depend upon what charge it is bearing. So this is in general, these two things you can remember. The mass of the ion is a lighter ions are deflected more obviously than the heavier ion, and the charge on the ions, which will more the charge, it will be again getting more the positive, more the charge, they will get more deflected. So less charge means +1 charge will have the less deflection. Then these deflected ions will be detected. So they will be detected by special detectors, which we'll be seeing again shortly. So they will be detected. So only ion stream B. So here, if in the diagram, if you see, this is the cursor which is moving here. This is the stream B. So stream B makes it right through the machine to the ion detector. So these ions are then detected, and then that will give you the signal. What is fragmentation process? So it actually, when we bombard the molecule, so look at the last equation. This is the molecular ion. First we bombard the molecule with the, say for example, 10 to 15 electron volt, where the ionization of that molecule will be there. So initially it should have been M, giving you M plus dot. That is what we call as a molecular ion. And then when you increase the energy of electron beam up to 70 electron volt, this molecular ion can further get fragmented. It will give you the daughter ions, where they can give you the cation radical or the neutral molecule. Okay, so and this process is known as fragmentation process. So these molecules, the daughter ions and also the molecular ions, they are very, very high energetic, and therefore they will further go fragmentation till they attain the stability. For stable ions, you get a better peaks, and unstable ions will not give the peak, and therefore it is possible sometimes that the molecular ion, they are so unstable that they further break up and they do not give you the peak for the molecular ion, or any such ion which is after fragmentation is not stable, will not show any peak or the stick bar on the graph. So we get, and here one more thing, neutral molecules which are there will not show any signal. Only the signal we get where we get the cation radical. So here, so uncharged free radicals do not show any signal, and they gets removed by the vacuum pump. So the vacuum is also applied there. Okay, and only the charged particles will be accelerated, deflected, and detected. Right. So now the mass spectrum, which is a plot of, so here is proper definition. It is a plot of relative current, which is taken as relative abundance or the relative intensity produced by ions versus the mass to charge ratio, which looks like a stick diagram. We will take one example here. Look at the structure here, what I've written for the hexane, N-hexane. So here, in this case, CH3, CH2, 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 CH3. These are the places everywhere it is quite possible that the fragmentation takes place. So first, what happens when you bombard with the electron, it will first knock off one electron and it will give you the molecular ion. And here, if you count the total number of carbon atoms, 6 carbon, and total there are 14 hydrogens. So the molecular weight for the N-hexane is 86. So we get a first peak at 86. So here you see the molecular ion peak is at 86. Okay, and then this further can undergo fragmentation. Different ways of fragmentations are there. I'll just use this. Yeah, I've just come out of this mode because I want to show you something. Yeah, so here the molecule which can, just a minute. Yeah, so the molecule N-hexane can undergo the fragmentation at different places. So this can get fragmented in this way or the other way or the third way, fourth way, fifth way. So anywhere, all these fragmentations you see are possible. If it goes like first methyl group goes, you will get the first peak at 71 because the methyl group is deleted. Here suppose it undergoes like this was A, B, C, D if I write down. So if it undergoes like loss of ethyl group, then you will get a peak at 57. So this is the 57, which is the most stable one. So you get a stronger peak there. You see the intensity or the abundance of that particular peak is more than the other. Okay, there is a possibility that in between the it breaks at, say for example, the third one between the two carbons, giving you two N-propylium ions, so with mass to charge ratio with the 43, or it is possible that the here in the second one you see the ethyl group which was there as a radical, and the remaining part with the 4 carbon atom was with a cation. If it happens other way, that is ethyl group is a cation and the other part becomes radical, then you get a signal at 29. And then same as the first one, reverse of that, similar to first one, reverse of that, we can get a signal at 15. So here for N-hexane, the fragmentations what we get is the 86 is for the molecular ion peak, 57 is for the first fragment when the methyl group gets knocked off, then the 43, 29 and 15. So here we get the different peaks at different level, just because every time some part is getting fragmented and we get the next molecule. Okay, so here we get the mass spectrometer instrument we will go for in detail. So mass spectrometer, what we have is mainly built up of 5 parts. Actually, it is the inlet what we have, which is used for the introducing the sample from the ambient room temperature into the ion source. We have the second one as ion source, where the converting sample molecules to ions. Then we have a mass analyzer separating ions according to their mass. Then we have a detector which detects the ion, and the recorder which will record the signals and then give you amplify the signals and give you the mass spectrum. Okay, and this all is done under the high vacuum system. So what is the high vacuum system? Almost 10 to the minus 6 to 10 to the minus 9 torr is that much vacuum is applied, for which they use turbo pumps, diffusion pumps, rough pumps, rotary pumps, like that. Okay, the main part is your inlet. That is where the samples are actually introduced into the mass spectrometer. So that can have the simple sample plate or the target, or also the combination of other methods, chromatographic method like GC, HPLC, or can have the solid probe. Okay, we'll see in detail. Ion source are different ion source which we use for the ionization. Can be EI, electron impact, or the CI, chemical impact, or the ESI, MALDI, ion spray, FAB, LS, MS, etc. Mass filters. So after ionization, the ion gets transferred to the analyzer or the filter. Different filters are there. We are going to discuss in detail. That is the like TOF, time of flight, quadrupole, ion trap, magnetic sector, FTMS, etc. Detectors are like microch plate, electron multiplier, hybrid detectors also used, and then we get a signal that is out that as a spectrum which can be recorded. Of course, it is now a computerized thing, so we get a very good signals there. So this is the whole instrument. The physics behind this actually is something like charge particle passing through a magnetic field is deflected along a circular path on the radius that is proportional to the mass to charge ratio. So here it is actually looking like this, the signals where the light source is used, and then it passes through here, and then ionization chamber is there, acceleration plate is there, and here is the curvature you see where the beams, the electrons, the atoms or the charge particle gets deflected, and then they are recorded to give you the spectrum which looks like this. So sample inlet, what are the common inlet systems? What we have is the batch inlet, or the direct probe, or the chromatographic, as I said, or the syringe like in ESI we will see, and the multi probes are also there. Solid, liquid, or the gaseous form, all 3 forms can be introduced in 3 different ways depending upon which instrument we are using. So first is ionization. What ionization does? We say the ionization is actually going to ionize the sample, the atom. Okay, and here we have the different methods like electron impact or the fast atom bombardment system, electron spray system, or the matrix assisted laser desorption, in short, which is called as MALDI, which are used for this. So again, which phase we use it here? The gas phase, if you use it, generally electron ionization, EI, CI, FI, these methods are commonly used, and out of that also you will find the electron ionization and the CI. So these methods are more commonly used, and because in electron ionization we use the energetic electron, for the chemical we use the reagent, which is the gaseous ions. So all these things again we are going to see in detail. If you are really want to appear for your exam also after this FDP, you can take the screenshot of this. This is going to be important for one to know, not for this exam of course, but it is also useful for one to understand what is ionization, how does it take place, and what is the ionizing agent actually. So these are the different ionization processes what we use. We also use the desorption methods, where different desorption methods like FD, PD, FAB, MALDI, etc. are used. So here are some soft techniques and the hard techniques which are there, like electron ionization, EI and CI are considered to be hard techniques, whereas MALDI, etc. are used for as a soft techniques. And generally soft techniques are better for the biological molecules, and for the organic compounds we use the hard gas phase ionization techniques. So it depends upon which molecule you want to analyze, which molecule you want to interpret its structure. So let's go one by one. Let's see the first, which is most commonly used, widely used for all the organic chemists, by all the organic chemists, is electron impact ionization. What happens here is you see that the electron which is there, here is bombarded on the molecule. It loses the electrons again further, maybe one electron to give you the molecular ion, which is most of the time for the organic compounds it is stable, except few examples, we'll see that in detail. So then it can have the it can further go into the fragmentation process. So here this is the mass analyte, and these are under the magnet, high energy electron, almost 70 electron volt is the energy which is used for generation of this molecular ion, and then that passes to the mass analyzer. So very high energetic electrons are bombarded. So what happens here, we said already that the molecular ion which is formed is undergoes the ionization and then the fragmentation. It will give you the fragment molecule, as one of them may be the cation radical and another is the simple radical, which is a neutral molecule which is not going to give you the signal and will be taken out. What is the advantage of the EI system? Is the fragmentation we get very good. We get a very good fragmentation, and then it gives you the structural information. And very important thing is these are reproducible results what we get. The disadvantages of EI is you should have the thermally stable and the volatile compound. And even only the small compounds are some small amount are of the sample is ionized and unstable for the molecular ions. So as we said, the M plus dot which is there molecular ion, if this is generally unstable, so generally we get a very small peak or the bar for the molecular ion peak, and therefore sometimes may be absent for some compounds, and we generally get the fragment molecule like M2, M3, etc. So we get the daughter ions more there. Okay, then what we get have the second ionization method, what we call as a chemical ionization method. In this case, the molecules are not directly bombarded by the electron, rather there is some other gas, reagent gas we call it as. So there is some other reagent gas which is used, which is actually first charged, which is actually first ionized, and that is then that further it will attack on the compound. So softer than the EI method, what we call it as. So this is in the two step, as we said. The first step is reagent gas is ionized, and commonly used reagent gas are like methane, tertiary butane, ammonia, nitrous acid, oxygen and hydrogen. And then in the second step, these reagent ions then attack on the gets bombarded on the sample molecule and to form the molecular ion. So this is generally definitely better than EI, but depends on what molecules you want to use it. Okay, so two types of CI, we get a positive chemical ionization and negative. Negative ions are generally not useful for us much, but counter ion of that, obviously a positive chemical ionization, that is very important for us to get the signals. So what are the advantages for the CI chemical impact method? Is a high molecular weight compounds can be analyzed, which was not possible with the EI method, electron impact method. And in this case also the sample undergoes rapid fragmentation. Since we get a fragmentation is actually very important, because then joining these fragments will be something like jigsaw puzzle. You have to just join the fragments. We will see some interesting examples how to join them, and then we can get the information about the structure. But what are the limitations? It is again just like EI, it is not suitable for the thermally unstable compounds and the non-volatile compounds, and it is less sensitive than EI. So the next method which is used is the field ionization, where the strong fields are used, electric fields are used like this, and then that will ionize the molecule. So once you understand the CI and EI, you understood that first method for the mass spectroscopy is ionization. Different ways are used, like maybe the high energetic electron, or maybe some other chemical, or maybe the electric fields are used. So different ways are there whereby we can ionize the molecule. The advantages are there. This is definitely softer than the EI and CI, but the problem here is it is having the low resolution and not suitable for again the thermally unstable or the non-volatile compound, and again it is less sensitive than the EI method. And no fragmentation occurs, or little fragmentation occurs, so we get not an important structural information. And next comes the field desorption method, where it is actually similar to FI only, field ionization only, but in this case we use the whiskers and ionize by applying the high voltage to the emitter, and useful. Now this is useful for the non-volatile. Field ionization was not much useful for the non-volatile compound. So you see every day by day all the different new inventions are there which will make it better and better. So it is good for the small molecules and like the molecular weight of polymers and petrochemicals. But of course, it is very sensitive to alkali metals. Then sample must be soluble in solvent and not suitable for the thermally unstable compounds, and even the little fragmentation here also it comes. So when you compare the FI and FD, here you will find the in FI the sample is heated in vacuum, and here it is directly placed on the surface, whereas in FI it is with the volatile and the thermally stable compounds we need to have. Here we can use the non-volatile or the thermally labile compound. And there is a very famous another method, that is ESI, electrospray ionization, which is can be used for the polymers or the proteins, for the compound which is having the molecular weight more than even 10,000 also. So in this case what they do is they use this, consist of very fine spray, fine needles and the series of skimmers what we use, and then they're creating a fine spray of the charged droplet. So here you see the fine spray of the charged droplet. So here also this helps in ionization. Then what we have is the electrospray ionization, dissolving capillary, and the warm nitrogen repeat itself free from, and this is this is the word what I wanted to use, that is a Coulombic fission takes place there. Advantages are high molecular weight compounds can be ionized with this, and it can be used along with the LC or the HPLC at the capillary electrophoresis. We have very recent ones are like plasma desorption method, where they use the californium-252 as an energetic fragment to create the energetic fragment. Very highly exothermic nucleus it has. So here what they do is the plasma zones are dissolved. So they spray a plasma here, and then the ions are dissolved from this, and then ionize the target molecule. Then we have a laser desorption, similar to the plasma desorption method for ionization. This is another very famous one, which is most of the time used by the biologists, and that is known as MALDI, which is a matrix assisted laser desorption ionization. It actually takes place in 2 steps. The first is the matrix, which is actually ionized, and then this ionized matrix is then exposed to the laser. So matrix absorbs the laser energy, transfers some of its energy to the analyte molecule, and then this analyte molecule gets ionized, and then we get the ionization done for that, when the further process which will be similar. So these are the different ionization methods. What are the advantages? Of course, we said soft ionization, and therefore it is used for many biologists. Then we have the complex mixture can be done, sensitivity can be there, then atmole sensitivity is possible, and almost more than molecular weight, that is more than the 3 lakhs Dalton, that is Dalton is a unit of polymer. How many monomers are there? I think molecular weight of that. So that much of the compounds having the molecular weight can also be ionized with this, can be ionized with this. But disadvantage is, of course, the softer the method, low resolution we will get, and availability is also possible. We have another FAB method, that is fast atom bombardment, where also the ionization which is done liquid secondary ion mass spectrometry, what we call it as for this. Then we have the APCI, where the atmospheric pressure chemical ionization is there, where there is a corona discharge which is there, and that will give you the ionization. A few more are there, secondary ion mass spectrometry, SIMS are there. So these are the different methods which were used for the ionization. Once the molecule is ionized and maybe fragmented, it is allowed to pass through the analyzer. So it goes to the mass analyzer. Different mass analyzers are there, like we have the very famous one like a quadrupole and TOF. These are the one which are quite used, but there are other also like ion trap and the magnetic sector also used. The latest one is the FTICR, that is ion cyclotron analyzer, but very costly, but of course very useful, because it is going to give you the highest resolution exact mass, but it is very costly. Okay, so commonly used one are of course the quadrupole and the time of flight machines are there. So what is quadrupole is, as the name suggests, quadrupole is the one where we have the 4 units, 4 parallel cylindrical rods are there. As you can see this figure, 4 cylindrical rods are there, with 2 of them are with a positive charge and 2 of them with a negative charge are there. So ions travel between these rods, and only the ions with the specific mass to charge ratio will exit the quadrupole, and other ions will collide the rod. So this is the analyzer. These are the analyzers which will actually give you will sort these different types of ions and will send this to the detector. We have the time of flight analyzer, so TOF, commonly known as, which is actually used without the electric or magnetic field. So here the separation is based on the kinetic energy and the velocity of ions. So the time that is subsequently taken for the ions to reach the detector is known as at the known distance is measured in this case actually. So in this case the flight time is measured correlated with the mass to charge ratio. So that is the way we get the analyzer. So then we have an ion trap. As the name suggests, the ions are getting trapped into this, which is like you see the 2 cups are there, inverted cups are there. They are where the ions can be trapped. So this is the another ion trap mass analyzer. We have a FTICR, that is a cyclotron resonance, which was developed in Canada 1974, which is a miniature of cyclotron, but since it becomes FT, Fourier transform, it allows many ions masses to be determined simultaneously, and therefore very interesting, very good for mass analyzer, and very good results we get out of this. So higher mass resolution we can get with the FTICR. Then we have a magnetic sector analyzer, where also the TOF, similar to TOF we have, where the ions are accelerated to the flight tube. Then comes the flight detectors. So different types of mass detectors are used. Mass detectors like Faraday cup, electron multimeter, photomultiplier, convergent diode, array detector, high energy diode, or the charge detector are used. So these detectors, what they will do is they will detect the current signal generated from the incident ion. Different mass detectors are used to detect the ions depending on the type of mass spectrometer. So it depends upon what you want to, which sample you want to analyze. There are different detectors can be taken. Okay, so Faraday cup means what? It is a metal cup which is placed in the path of ion beams and is attached to an electrometer which measures the ion beam current. And so it can be less sensitive. Then we have the electron multiplier tube, which is similar to your photomultiplier tube, very commonly analytical chemistry, people know about this, which consists of the series of diodes that eject secondary electron when they are struck by ion. Then we have a channeltron, which is a horn-shaped continuous diode structure that is coated on the inside with the electron emissive material. We have a Dalley detector, which consists of metal knob that emits the secondary electrons. So here is the comparison for the mass detectors. What are the advantages and disadvantages? Every detector has its own advantages, but may have some disadvantages, which becomes the advantage of, which can be overcome by the advantage of second one. So like in Faraday cup, we have a low amplification, but if you see the another one, for example, array type of detector, which is fast and sensitive. So all these will have their own advantages and disadvantages. Finally, when after the detection is done, we get a data interpretation. We can go for the data. What is the data comes out of it? We can get a spectrum of it. So mass difference between the consecutive ions within the series allows the identity of the fragment. So earlier also we saw one example. So here is another example. Say for example, 2-butanone is a compound where the molecular formula of that is what we say C4H, how much? 8 and O, oxygen is there. So with the C4H8O, if you see the last part, what we get, the last bar, what we get is nothing but 99% times, of course, it is a molecular ion peak. It corresponds to the total molecular weight of the compound. So, for example, if you see the ethane molecule will have if it knocks off one electron, one proton, it gives you the molecular ion peak at 29. Then what is fragment ion? Once this molecular ion undergoes, it itself is highly energetic, it will undergo the fragmentation. In this fragmentation, you can get the daughter ions as M1 and M2. For example, here, the methanol, which is having the M by Z as the 32, can get rid of one proton here and can give you the molecular ion, the fragment ion at 31. So losing one H dot. So if you see from the methanol molecular ion peak 32, if you lose one hydrogen with one electron, then you will get CH2OH, which is with the M by Z as 31. There can be a rearranged ion, where the original molecule is not the same, directly undergoing fragmentation. It could be other way also, it can undergo the fragmentation. So how it can undergo? It is by rearranging the molecule. Why rearrangement takes place? Rearrangement will take place for the sake of stability. As I said, is the stable ions are only going to be detected more, and therefore the rearrangement in some cases takes place. So we will see also what different types of rearrangements are there. Then there can be, as I said, is multi-charge ions, like it can have the charge +2 or +3. There can be a negative ions also. And this is a very important one, which is what we call as a metastable ion. What happens here, if you look at this figure, here is the ion source. From here, the compound enters, and now the ionized M+, which is ionized already, can enter in this deflection area. Okay, so in this case, what happens in during this, when it enters from here, it is supposed to go to the detector directly. But sometimes it is possible before it passes through the detector, this molecular ion can further fragment in this particular area. So this particular area will give you the M2, which is actually going to the detector. Do you understand? Like the molecular ion which was supposed to go to the detector, or the fragment ions from this ion source were supposed to go to the detector, can further undergo fragmentation in this area, where now it is neither the M1 nor the M2. Okay, it will form the M2, but it is not of this because it was not formed earlier. It is not a pure M2. And therefore, these type of ions will be known as metastable ion. So if you read this, M1 may decompose to M2 while being accelerated. The resultant daughter ion M2 will not be recorded either as M1 or M2, but at the position which is known as M star, and will give you the poor peak actually. Okay, so you will see the metastable ions will not give you the sharp bar, but it will give you a poor peak like this. Okay, so they are denoted with the M star, and that can be calculated also. Like if you get a peak at M star value, and you know that you cannot assign directly what it is, so that can be calculated by having the formula as mx square upon the mm. So mx means the mass of daughter ion, that is M2 we talked at, and mm is the mass of the molecular ion, that is M1 what we said. Okay, let's take example like acetophenone with the M by Z 120 is the compound which will fragment at 105. We will see this again, one example in detail afterwards. So it will get fragmented as M by Z 105 with the loss of methyl group. So acetophenone, as you know, the structure has the benzene ring, C double bond O and CH3. So in this case, we have the loss of this methyl group. So what remains here is the phenyl ring and the C double bond O, which has M by Z value as 105. So 105 directly, it is, if it is going to give you the signal, it will come at 105. But in between, in this acceleration region, suppose it gets further fragmented, like then it will give you the signal which is of M2, but it is not pure M2. So that will give you the signal at as a metastable ion, which can be calculated. So you will get a signal at 91. Okay, now getting a signal at 91 indicates this is not a pure fragment of acetophenone, and that indicates must be the fragmentation pattern is something different, and therefore you can understand the CO is also going, and this CO is going somewhere in between, and therefore we are not getting exactly 105, neither the 77 for phenyl ring. Okay, we will see again that in detail example. Okay, so what is the nature of metastable ion? They have the low kinetic energy than normal ions, and therefore the peaks are smaller and also the broader. So you don't get a bar peak, but we get a smaller and the broader peak there. And then what is the significance? As I said, it is useful in helping to establish the fragment routes and also can distinguish between the fragmentation process. Very, very important information you get from the metastable ions. So general rules of fragmentation. What are, how do you think, what should I, how should I fragment it? So if you look at this, if you look at this particular structure here, you can see there are different ways, like you see 4 carbon atoms are there, and each carbon is attached with 1 or 2 or 3 hydrogen atoms. In the first case, it is attached to 2 hydrogen atoms, but 1 double bond. In the second, one hydrogen atom. In the third case, 2 hydrogen, but with all single bonds. In the fourth case, it is all 3 hydrogen atoms. So now when you want to think of fragmentation pattern, there are variety of ways. The variety of ways as you can have the fragmentation of this CH, we can have the fragmentation at this CH, we can have the fragmentation at this CH, CC bond or this CC bond. So like that, you can have the different ways of fragmentation. So the relative height of this molecular peak is greatest for the straight chain compound, and it decreases as the degree of branching increases. This is the first rule one must remember. And then the relative height of the molecular peak generally decreases with the increasing molecular weight. The very important one is cleavage is favored as it gives you the the stable species, and therefore, as you all know that the carbocations, tertiary carbocations are more stable than the secondary, which are more stable than primary than the methyl carbocations. So we see depending upon what stability is going to happen, accordingly it will undergo the fragmentation. So the most stable fragment will be more preferred. So here, if you look at this particular example, double bonded compound, you will see if it, and there are different ways I have shown, but if it undergoes a fragmentation in this manner, where the methyl group is preferably lost, it is going to give you a allylic carbocation. So here the allylic carbocation means a carbocation having its adjacent with the double bond, it can undergo the resonance. It can undergo the resonance, and therefore it is going to be more stable. So in this case, we can have the fragmentation in this pattern. So one must understand how the fragmentation takes place. It depends upon the stability of the compound. So then we have the saturated rings. Saturated rings can lose the alkyl side. So suppose here you have a ring and here you have a side chain. So preferably it will undergo the alpha carbon. So this here it is preferred, it is breaking, and so that here it will generate a secondary carbocation. In case of suppose you have aromatic compounds, then depending upon what is the nature of the side chain, it may undergo some rearrangement or it may undergo the fragmentation. Preferably it will go for this species where the one CH2 is retained, and that is what we say methylinium benzene, what they have to give you 7 carbon atom tropylium species, which could be a cyclohepta tetranyl cation. Trinyl cation. Okay, so here we can have the species which is at molecular weight M by Z value at 91 is going to be the most stable one. So as we said, double bonds will favor allylic cleavage here, and for having the allylic carbocation, and then the elimination. Yeah, this is also very important one. It will prefer, the elimination will prefer the elimination of small and stable neutral molecules. So, you know, different ways of fragmentations are there. So Stevenson's rule has been given, which states that when an ion fragments, the positive charge will remain on the fragment of lowest ionization potential. So it has lowest ionization potential, that is more stable. So here the positive charge, when you see the cleavage of 1-methyl pentane, there are different ways, like it could have broken in this way or you know, different ways, but it prefers to break in to give you mainly these two parts, where the positive charge will be here, because you see this is a secondary carbocation and it is going to be a more stable fragment. So all these fragmentations will be, you know, general rule can be, one can remember. Different modes of fragmentation, how it will break. As we have learned already few of the methods, like what do you mean by homolytic and all those things, we will see systematically that there are mainly two modes of cleavage are there. Simple cleavage and sometimes rearrangement can be there. Simple cleavage, any beginner can understand. Yes. Okay, so simple cleavage any beginner can understand. It can take place with the homolytic cleavage, heterolytic, or even the tetradienzyl reaction. So all these are very simple to understand. But suppose the rearrangement takes place, which are of different types, then one can, you know, have to be expert enough to analyze the spectrum perfectly. Simple cleavage like homolytic means one by one every part will go off. Like here, maybe the first the R goes, then CH2 goes, then another CH2 goes. So like sequentially it can break, or it may break here directly in between. So to give you two species like this. So it depends upon the molecule and the stability of that cation which is formed. Heterolytic means only one of them will take both electron toward itself, and like here in CH bromo butane, you can have the bromine with the free radical, and then the four carbon atom species can be with the cation. Carbon cation can be there, or it can be a heterodyl solder. Everybody knows what is diol solder, where the diene reacts with the dienophile, and we'll give you a cyclic structure. Here, what can happen if you inject the compound like cyclohexene, it can undergo the retrodiol solder, where opposite of the mechanism takes place and can give you the stable small molecule which is going out. We already said that the breaking prefers to give you the smaller molecular neutral ion, stable neutral ion. So here the cyclohexene, when we see, it will eliminate this ethene molecule, and you will get a very strong peak for the cyclobutadiene. But when it comes to the rearrangement, as we said, there are experts need to be there to interpret the data. So here different ways what can happen is scrambling, where the bond will just rearrange itself, give you a stable carbocation. The very important one rearrangement out of this is known as McLafferty rearrangement, where the fragmentation due to the rearrangement of the molecular ion peak is there. You can understand better with the next example. If you look at this, the parent ion, here the parent ion is suppose the ketone. Now look at the structure of this ketone. There is this particular is a functional group. This is your alpha carbon, beta carbon, and gamma carbon. If this gamma carbon has a gamma hydrogen, which is in perfect position with this carbonyl oxygen, then the rearrangement takes place like this. It will form a 6-member stable transition state, and will get rearranged to give you the two species. One is the alkene and one is the ketone only. It is an enol form of this ketone, so you get an enol form. Okay, so McLafferty is many times is possible in the mass spectrometer, preferably when there is a presence of gamma carbonyl group with the gamma hydrogen. Okay, and this can be with the ketone, this can be with the acid, this can be with the ester. With the ester, you see, please have a look at this particular last two figures. Here I have taken COOR. So here my gamma part is into the carboxylate part. So here is the gamma hydrogen. So McLafferty with that also is possible. Now in the second example, you see the gamma hydrogen is present with this oxy part, the alkoxy part which is there in this ester. So McLafferty is possible with that also. So that is the beauty of McLafferty rearrangement. One must understand that it will undergo the rearrangement whenever there is a carbonyl group, and it will have the whenever there is a presence of gamma hydrogen, one of the very important rearrangements in the mass spectroscopy. Then there can be elimination where the again to give you the good stable carbocation, like as we said in alcohols, here it will undergo the elimination of water, and therefore this for the alcohol we should have expected the peak at say for X molecular weight, but we never see the X molecular weight peak, but we will see always the loss of 18 mass, because it will lose a molecule of water, that is it will lose immediately the OH and H as a water, and you will always get the X minus 18 as molecular ion peak. And that's the speciality of alcohol peaks, that is alcohol will generally not give you the signal at molecular ion peak, it will always show the molecular ion peak at 18 less. Then we have the fragmentation adjacent to the branching. How do you get it? More the branching we have, this branching will help to give you the stable carbocation, and therefore that type of elimination also there. We can have the hydrogen transfer. So like here you can see the hydrogen gets transferred from this ketone molecule to the immediate ester, the ester molecule to this, giving you the phenolic compound, and then the loss of the ketene type of species. Then we have the ortho effect, what we call as a for the with the substituted aromatics. When you have the ortho position, a group such that it can undergo the rearrangement, it will give you the ortho effect also. So these are the different rearrangements which can happen in the mass spectrometer, and there you need to understand these analytes also, these signals also, to when you are joining this jigsaw puzzle. Another important thing to remember when you are interpreting the mass spectrum, that is known as isotope effect. So when you go for the isotope effect, that is, let's see the example. Bromine, brominated compound. Bromine is having the isotopes as 79 Br and the 81 Br, which is in the natural abundance for both is 1:1. So when you take, let's take the simple example, methyl bromide. Methyl bromide, methyl group is there. That means 1 carbon, 3 hydrogen, which makes it 15 as a mass number for the methyl group. And the Br is 79 or 81. When I add 79 here, it gives me the total of 4 and 9. 94. And if I add 81 into that, it will give you 96. So here you see the two peaks with the 1:1 ratio as 94 and 96 has appeared. This indicates that there is a Br present, because the Br has an isotope with the M and M+2. These peaks we get. If you get M peak and M+2 peak with the 1:1 ratio, then one can imagine there is a presence of Br. Okay, and since we say that there is a presence of Br, the next fragment you can see the CH3Br will lose the methyl group, that is 15, which is coming here, and 79 and 81 remains here. So it is very easy to interpret once you understand this isotope pattern. Very important is you can imagine what we can see, where we are getting the peaks, what is the, how it is looking like, what is the ratio, what is the difference between the two peaks. All this will give you valuable information when you are interpreting the signals. Okay, so here in similar to this bromobenzene, you will find the molecular ion peak at 156, and directly you will get 77. 77 is for the phenyl ring, C6H7. Okay, C6H5. That is 77. It will come. Okay, so 12 6 are 72 plus 5 is 77. So for the phenyl ring, you will get a 77 peak, and the additional we have the bromine one, that is 79 and 81. So here for chlorine, now you see chlorine is actually present as chlorine 35 and chlorine 37, but the relative abundance for the chlorine is 3:1. So you will find, look at this molecular ion peak, you will find the signal at M plus and M plus for +2. M means like 35 chlorine and the compound having 37 chlorine. Look at the height, they are 1:3 or 3:1 ratio. They are in the height of 3:1. So when this molecule will lose one of the methyl group, that is 15 lost, it will give you the fragment 1 as 63 and 65, still having the Cl in that. Suppose the directly Cl goes off, you will get somewhere here is a 43 peak, which is actually obtained by, I will write it here, CH3, CH and CH3 and Cl. So if it loses this Cl part, if it loses the Cl part, this part which remains is 43. So here you are getting the base peak at 43. Can you see this? Okay, you are getting the base peak at 43. So this 43 was because of the presence of the loss of the Cl. So here you will see a single peak. You are not going to get 2 peaks here. Sorry, not here. Here you will get a single peak, not a 2 peaks in 3:1 ratio. Further, this fragment, which is at I think 28, that is because now the another methyl group has also gone. So this is the way how the fragmentation pattern has to be understood, how it takes place. So these are the some isotopes and their abundances which are there. So mainly important for us is the carbon, which is having 12, and we do have C13 also. So C13 abundance is just 1, and therefore most of the time you will see, I don't know whether it can be seen here, no, not in this, but there are some very small there are some very small peaks here with for the C13 carbon are also there. For NMR we have altogether C13 NMR, but here with the help of abundance we can also find out the C13 elements which are there. So here the main important thing is for us, it is the carbon which is 99:1 ratio, for chlorine it is 3:1 ratio, and for the bromine it is almost 1:1 ratio. So depending upon which peak you are getting and where you are getting, so for C13 you will get a peak at M plus value and M plus 1 value here with the ratio 99:1. Whereas for the chlorinated compound, you will get the ratio in 3:1. For the brominated compound, you will get the M and M plus 2 values at 1:1 ratio. So with the help of mass spectrum, one can immediately understand which element is present. One more important rule for one must understand when he is interpreting the mass spectrum as the nitrogen rule. The nitrogen rule says that when you have an even molecular weight for the final compound, like here, the M plus ion is 89. Okay, when you have the even one, then you can think of there is no nitrogen present. I repeat, if the even molecular weight is there for the molecular ion peak, means it contains no nitrogen or it contains even number of nitrogen atoms. In other words, it is a odd molecular weight. Okay, so odd molecular weight when you get for the M plus ion, that indicate there is a presence of nitrogen. Maybe one nitrogen or maybe odd number of nitrogens is present. But when you get even number of molecular ion peak, then you will definitely have the absence of nitrogen atom or you will have the even number of nitrogen atom in that. One more thing to see at this, the fragment may be even or odd, doesn't depend upon, it does not depend upon the nitrogen. Only we are talking about the molecular ion peak. We have to look at where we are getting the molecular ion peak, whether it is at even position or odd position, and depending upon that you can interpret whether the nitrogen is present, whether it is odd or even, or it is absent, or all this information you can get from the nitrogen rule. So then we'll go for the general fragmentation schemes. So we can have the weakly bonds will break easily. Then we have the tendency for the formation of more stable fragment ion, and then the more fragmentation process takes place by the imaginary cyclic transition state. All these things we already saw. So common cleavage process also we saw it here. Okay, so now we'll take the one example like fragmentation of 2,2-dimethyl butane. So as you can see, there are so many methyl groups are there, 1, 2, 3, and 4. Which one will prefer to undergo breaking? If you say this particular methyl group goes, in that case you will form the species which looks like CH3, CH3, CH3 and CH2 with the plus charge. That species will get. But this carbocation species is primary and it is going to be less stable, therefore it is less preferred. So first fragmentation may not be with this methyl group. The fragmentation may be two different ways. Either this methyl group will go or this methyl group will go. So not that, sorry. Or this ethyl group will go as a whole. Or the ethyl group will go. I repeat. Either this methyl group goes or the whole as a whole this ethyl group goes. Okay, so if the methyl group goes, look at this down, yes, here up. So methyl group is going, you create a stable tertiary carbocation and therefore you will get a signal at 71. Suppose the ethyl group goes, as you can see in the second fragmentation, you will generate a 57 as the peak there and the neutral fragment going as a ethyl group. So in short, with this example, you can understand which group is more preferred for the fragmentation. So in one example, what we saw is if it loses the last methyl group as carbon number 4, then it will generate a primary carbocation which is not stable, so therefore not preferred. It will prefer two ways, either the loss of yellow ethyl group or the loss of pink ethyl group. If it loses the methyl group, it will give you the mass at 71. a stable cation, or it loses the ethyl group, it will give the mass at 57, which is also another stable cation. In case of carbonyl group, it can give you, in case of carbonyl group, it will give you the stable resonance acylium cation. It will give you the stable acylium cation. Okay, so it depends upon which group is present, functional group is present in the compound. So here are few examples, like you can have a look, that is fragmentation of alkane, how it takes place. You can see how the breaking of this can go homologous way of this alkane molecule, since there is no problem of having the tertiary, formation of tertiary carbocation. One by one, every 15 molecule, every carbon can go off, and you can get, see, 86, 71. So with the loss of 15 means methyl group is going, then 57, 14. So every 14 units they are decreasing. That is for every CH2. So one methyl group is CH3 is 15, one CH2 group is 14. 12 carbon and 2 hydrogens means 14. Okay, so every first 15 and then every 14 is losing. So a very simple fragmentation, you can see that 86 giving you 71, 57, 43, 29, 15. So all these fragments can be seen. The moment you have a branch one, it will prefer to give you the stable carbocation. So therefore, you see the peak, here is 47. 43. In case of alkenes, we already saw it will give you the allylic cleavage to give you the stable carbocation. For aromatics, it will prefer, see here, the benzene is giving you 78, which is original is there, and by loss of one hydrogen, it will give you 77 for the phenyl one. But if you have the toluene, it will get rearranged to give you the, this is known as tropylium ion, and that peak you get a sharp peak at 91. Please remember this figure, we will be using shortly. Alcohols, you can see the primary alcohols will lose a molecule of water from this part, and therefore we'll see here no peak at 102, because it will always give you the peak here. You see, minus 18 peak we will get here. But that is the aliphatic alcohol. But if you have the aromatic alcohol or the primary alcohol, aliphatic one, which will not have the presence of beta hydrogen, then it will undergo the normal way of fragmentation. When you have the ethers, ether will give you the oxy radical, which will give you the major signal. For aldehydes, again, we can give see the signals. For carboxylic acid, esters, you see, we have already seen if that has the presence of gamma hydrogen, it will undergo the McLafferty rearrangement and will get the signal accordingly. Here is an interesting example. For amine, you see, all of them are C4H9NH2. All of them are butylamine. Okay, but all these butylamine, one is a primary, second one is a secondary one, and third one is a tertiary amine, what we have. Okay, primary amine is having only the one NH2, sorry, it is not a primary, secondary, tertiary amine. They all are primary amines, but attached to a primary carbon, secondary carbon, and tertiary carbon. Okay, you see the breaking is taking place in a different way. If it was attached to the primary carbon, it is giving you the base peak at 30. Okay, if it is the NH2 group is attached to the secondary carbon, it is giving you a peak at 44, and if it is attached to the tertiary carbon, it is giving you the peak at 58. So depending upon where this NH2 is attached, you will get a signal. One more thing I want you to observe, look at this molecular ion peak, it is odd. So do you remember nitrogen rule, what we saw? The nitrogen is present, so you are getting the molecular ion peak at odd number, that is 73. You can also distinguish between the two compounds. Like here you see, I have a pentane 2-one and pentane 3-one. If I subject these compounds, both these compounds in IR, I will hardly see the difference in their carbonyl frequency. Both of them will come almost at the same value, and I'll not be able to differentiate which is your 2-pentanone and which is your 3-pentanone. But if you take mass, you will immediately see if it is 2-pentanone, it will give you the signal at 43, because I already said is this part, CH3CO part is going to be the more stable one. Whereas if it is 3-pentanone, the COCH2CH3 part is going to be more stable and you will get a signal at 57. So immediately one can understand which one is 2-pentanone and which one is 3-pentanone. So we can use the mass spectroscopy for differentiation of the two compounds also. The very important one here is another interpretation based upon the mass spectrum is what we call as the rule of 13. Rule of 13 means, suppose you do not know the molecular formula. You all know that we determine the molecular formula based upon the percentage of carbon, hydrogen. So all that percentage analysis we find out, and then we get the empirical formula, and then we get the molecular formula. Suppose you don't have the method for doing it. Suppose you don't have the molecular formula with you. It will be, of course, very good if you have the molecular formula with you to solve the problem. But suppose you do not have the molecular formula with you, you can actually get the molecular formula. You can determine the formula given as M/13, which is therefore known as rule of 13. What is this 13 stands for? The 13 represents the total mass of one carbon and one hydrogen. You know that carbon is 12 and hydrogen is 1. So total makes 13. So you divide by 13. Whatever is your molecular ion peak you are getting, that you divide by 13, except that alcohol part, but you divide by 13. Whatever the numerator you get and whatever the remainder you will get. Okay, so you will get some values there, right? So the molecular formula for that is written as CN, where the N is the complete numerator what you get, and H is the numerator plus the remainder what you have. Okay, so that will give you total molecular formula as CnHn plus R. So this is the way you can justify, you can get the formula, molecular formula using the mass spectroscopy. Let's take one example. Suppose you have the mass spectrum giving you M plus value at 136. So when you divide 136, you will get 10 as the numerator and remaining is 6. Okay, and therefore when you look at the formula, I will write down C10 and H, N plus R means 16. So here you see it should have been, yeah, C10H6. So here you get the formula C10H16, and if you calculate the DBE, it is going to be different. Yeah, maybe the wrong one, I think. But if you just justify, this is not justifiable formula C10H6. Therefore, what we do is we can replace this one carbon or one hydrogen. One carbon is what, 12. So you can replace this by 12 hydrogens, and then we can just reduce the number of carbon atom to C9, and then you can get the hydrogens at 18. So this is the way we can use it. Maybe later we can have the other further examples. Okay, then we have the one again. If you just are interested in this mass spectroscopy, can take the this particular screenshot, because very important fragments you can understand. Like while solving the mass spectrum, you need not every time calculate there is 1 carbon and 3 hydrogen atoms. Immediately one must be able to say CH3 is losing means 15 molecular mass is losing. Ethyl group is losing means 29 is losing. Or if I say acidic group is going, then 45 is losing. For phenyl group, I should remember it is 77. For benzyl group, I should remember it is 91. For benzoyl one, I should remember it is 105. For where is this? Yeah, 43 is for the acetyl group. Or 57, these two 57 are important. That is for either butyl group, or I can have the propyloxy group here. Okay, so here, these are some fragments one can remember while solving the problems. Okay, let's solve one problem. I have given you 2 spectrum here. Okay, one spectrum has the molecular formula C8, H8 and O. Okay, if you are writing this, please look at this very carefully. It is the molecular formula C8H8O. Okay, and this has the molecular fragmentations at 120. This is your M plus ion peak, and you get the further fragments as 105, 77, 43, and 15. So these are your fragments. Okay, and this is the second compound, which also has the molecular formula 9H12 but has the M plus value at 120. Okay, now here I have given you the molecular formula C9H12 and C8H8O. Okay, but if this was not given, we would have used 120 divided by 13. So that should give me 9, and the remaining of that is how much it is? 16. Yes, so I would have got the formula as C9H16, and if you just maybe, yeah, so here 16, so here directly let me give you C8H8O. I have to interpret what is this coming at. So since both of them are giving me the value at 120, we'll use that C13 later, but here the 120, this further giving me 105. Both of them gives me the molecular formula at 120 only, but further fragments are different. 77, I told you to remember there may be a presence of phenyl ring. Okay, 43, if you remember having oxygen, so I can think of COCH3. Okay, so here if I look at the structure and try to solve like a jigsaw puzzle, I can have something like these fragments. The 15 is my methyl group, so I can have it like this. So if you look at all these fragments, I can collectively write down the structure as acetophenone, where this is my methyl group 15, C double bond O is there. This C double bond O may break in this fashion, or may break in another fashion to give me either this part as 105 or this part as 43. When this part as 105 is coming, this part becomes my 15. I hope you can understand the breaking. Okay, for the acetophenone, if my benzoyl group is going off, which makes a 105, my another part as 15 is coming, so I can assign my 15 and 105 part. Okay, if the breaking takes place in the other way around, that is the this way, in the other manner, then my 43 part is coming 1 and then 77 part, because the phenyl ring was 77 and this was 43. So I can see my another 77 part and 43 part is coming. So my total molecular formula was 120, giving me fractions at 105, 77, 43, 15. So I have to actually interpret based upon the previous one. These things, if I remember that generally 15 comes for what, 29 comes for what, 77 comes for what, 105, see last is 105 comes for what. I can immediately understand what are my fractions going to be. Okay, now if you see the second example, C9H12, then I can look at the fragments here, which are going to come only at 77 and 43. Of course, 120 is my molecular weight. Okay, so 77 if I get, I can imagine a phenyl ring. But if I see 43 was like in the previous example, I considered as acetyl group, CH3CO group. But in this case, molecular formula is not giving me oxygen. Okay, so since this is not talking about giving me oxygen, I cannot think that 43 is for my acetyl group. So if I go back here, I can see the 43 is coming for the propyl group. Okay, so I can think of the 43 group can be a presence of my propyl group. I can think of CH2, CH2, CH3. So you see, both of them were having the same molecular formula 120, but they were having different mass spectrum, and therefore I could interpret they are two different compounds. They are having different mass number, molecular formula, which could have been calculated with the help of rule of 13 also. Okay, and I can get based on the fragmentation, I can interpret which compound is present. Okay, so with that, we'll stop for the organic chemistry uses, and I quickly will go about the advanced mass spectroscopy method. I have just 5 minutes with me after I started my lecture. Okay, so now different types of mass spectroscopy is the what have advanced one, we have the GCMS, which is a combination of GC and mass spectroscopy. LCMS is a liquid chromatography, like what we say HPLC like type of things, can be coupled with the mass spectroscopy. There can be a tandem mass spectroscopy where MS are there, or we have LC, MS, or ESI, QT, QTOF, where the quadrupole mass filters and the TOF mass analyzers are used, and the multi are again can be coupled with the quadrupole and the TOF analyzer. So this is about the GCMS. What we have, some of you might have used also in this case the GC or LC, where the pre separation we use the GC, and post separation and after separation we use the mass spectroscopy. So GC, LCMS, we can have the HRMS. HRMS is like, will give you the high resolution, will give the masses in the almost up to the 4th and 5th digit also. So very, and nowadays no paper is accepted or simply on simple MS. They need all the HRMS data for this. Then there is a TARIS, that is thermal atomization resonance ionization spectroscopy. That is where the first the molecule gets ionized, atomized, and then it is getting ionized with the help of laser. So we get the thermal atomization and resonance ionization spectroscopy. So then we can have the ICMS, which is MS is coupled with the ion exchange, or sorry, ion chromatography. We can have the tandem mass spectroscopy, where the two mass spectrums are used. In the first case, first is utilized for purifies the analyte, and another analyzes the fragment. So that is the way it is used. Then we have the different configurations, and the tandem is mainly used for the biological molecules. Peptide fragmentation is there, then from that the sequencing can be done. Advantages are there definitely, but very, very costly. Look at the second last line. For one run you need 75,000 rupees. So that is really very costly, but yes, some people needs it for the biological analysis, so they are in use. We have also the tagged one, isotope tagged mass spectrometers, and then we have again 2D LC or LCMS type of things. We have the further ESI tandem, the multi-tandem we can have. And then finally, this is the mass spectrum of serotonin, which is actually responsible for your patience. So thank you very much for your patience.
Thank you, madam.
In time.
Okay, thank you, madam. It is a very informative talk given on the mass spectroscopy. And this mass spectroscopy, it is the technique that is used to measure the mass to charge ratio of the ions. And from this mass spectrum, we can identify unknown compound by using the molecular formula find out from this mass spectrum. Madam has explained in detail the history of the mass spectroscopy, basic theory, principles, scheme of the fragmentations, applications of the mass spectroscopy. Now I request to participant, the session, it is open for discussion. Any doubt or any query, you can ask to any question can ask. Session is open for question answer.
Ma'am, if we have nanoparticles of zinc, can we characterize by this, ma'am?
I actually personally never used, because for organic chemistry I've done a lot of work in mass spectroscopy. I'm not sure about the inorganic compounds. Maybe it is useful, because mass spectroscopy is used for the nanoparticles, but I cannot tell you all in details.
No, ZNO cannot be analyzed by the MS. If ZNO is dissolved, then you can analyze the zinc for isotope or the for concentration, but ZNO cannot be.
Thank you, sir.
And if ZNO is associated with proteins, can we do it by GCMS, ma'am?
We will just fragment that, I think, with the GC. For the proteins, if you are just fragmenting that, we'll get a lot of fragments. But yes, for organic part, we can do it with it. As sir said, we will not be able to do with the ZN.
Okay. Thank you, ma'am.
Any other participant?
So may I propose vote of thanks?
Okay, no question. I am request to Dr. Mujab Sheikh give vote of thanks.
On behalf of Krishna More College, Akode, MHRD, Savitribai Pune University, and on behalf of all the participants of technical research in clinical and biological sciences FDP, it is my privilege to give a vote of thanks to Dr. Sangeeta V. Jagtap ma'am. First of all, I was very interested in mass spectroscopy. I belong to actually to biological sciences, and this mass spectroscopy is not that much known as compared to that of the chemistry people. Is not been known to us, but I was very much enthusiastic in knowing the process and procedure and its application. So that's why I joined this FDP, and after listening to the wonderful lecture delivered by the ma'am, so the way she had started her lecture from the basic and its application and its uses for the chemical science students as well as biological science students, the way she had presented in the slides and with the examples, so it was so interesting. And then she explains with lot of examples like preparation of molecular ions fragmentation, and how to analyze the compounds, and how to take the peak and what not to consider as a basic peak, and how to correlate the peaks with the different compounds. And in a way, she has continued her lecture that it creates another interest to know further and further what next will be coming in her slides. So she was explaining in such a way that what we all were, I'm sure that what we all were expecting out of this particular lecture, we have been delivered the very same one. So at the last as well, so determination of molecular formula, that was a kind of like a new learning. Means we were having a kind of little bit knowing that molecular formula is there, but how exactly one can be identified by using the mass spectroscope, that is clear to the beautiful explanation by the ma'am. So once again, on behalf of all of our participants, I, Dr. Sheikh Mujib, take this privilege to thank Dr. Sangeeta V. ma'am. Thank you very much, ma'am, for such a wonderful lecture.
Thank you, Mujib Sheikh from Pune College, giving the vote of thanks for this session to Jagtap madam. Now, I also interested for this mass spectroscopy because we are basically young people, and we are the colleagues here about 25 years in Babarao Developed College, Sagvit. And we try to synthesize these organic compounds and new organic molecules, and for that purpose, we carried out the mass spectrum. Now to talk on mass spectroscopy, and our delegates or participants are enjoy this lecture, and he all know this concept, the small things regarding this mass spectroscopy.
Thank you. It was very exhaustive. I hope I could deliver everything because it was very vast subject. So I just touched upon all the aspects, right from the history to the advanced thing, but everything in detail definitely needs more time actually. So it was 1.5 lecture, hour lecture. So I had to manage all the things. So
FDP, therefore, life sciences teachers also involved in that FDP. Therefore, also they know the better. They also understand some things from this master.
I try to cover the instrumentation and organic chemistry part more than the biological part.
Yes. Therefore, biological teachers also try to this master interpretation also make easy by this lecture. I hope. Okay. Now we move to next session. Next session is again a technical session, demonstration session regarding the instrumentation part. Now I request Dr. D.R. Sinde. Today's session we discuss about how to prepare the nanoparticles. We are basically solid state chemistry people, and we always make different nanoparticles and we use it in different applications. Therefore, a number of times, as again I collaborate with the organic people for this purpose of whatever nanoparticles we synthesize in the laboratory for the purpose of its application in terms of the catalysis or in terms of the various applications or biological evaluation also there. Therefore, very important things about that, the nanoparticles are the tremendous application of the various fields. Not only in the biological field, also it can be the medicines, or it is also in catalysis and not only in the catalysis, different applications for that nanoparticle. Now, today's session, Sindhu sir try to explain how to prepare nanoparticle by using the chemical method and as also use the biological methods. Therefore, by using the biological template, we also try to prepare the synthesis in ZNO nanoparticles, and by using the chemical route, very simple mechanochemical route, or sometimes we also use the co-precipitation method. Different methods are there. The colloidal nanoparticle we prepared by this using the different chemical technique. So I request our colleagues, Dr. D.R. Sinde, please take the charge now of this session and explain simple methods of synthesis of silver nanoparticle by both biological as well as chemical. And it's a simple characterization technique. They like visible spectrophotometer. By using the visible spectrophotometer, we can characterize these whatever nanoparticles are synthesized or not. Okay, over to Sinde sir. Are you ready, Sinde sir?
Yes, I am ready.
Okay, okay. Take charge now.
Yeah. All the participants, yesterday we have seen the hands-on training and determination of the
Let us here may I take charge, guys. Cooperate with.
Okay, so today's session is the simple green method of synthesis of silver nanoparticle of different size and the study of their band gap. As well as if time permits, I try to represent how copper oxide nanoparticles they are obtained and how they can be synthesized. So particularly today, why I have selected the silver nanoparticle, one thing. And second one is copper oxide nanoparticle. Both these are the nanomaterials which are biologically active and have number of applications. You just put up the word in the Google search. I will not give the application. As a technical session, I cannot give. And you have a Google Assistant of the Google search. Okay, you just search the application of silver nanoparticle. They are in the environmental remediation, analytical chemistry, in the medical field, and the variety. Okay, they are antifungal properties, they have antibacterial properties, they can be used for the cancer treatment. Number of applications of silver nanoparticle are there. They can be used for the water purification. Same is the case with copper oxide. Copper oxide nanoparticle, they are biologically active. They are having antifungal activity, antibacterial activity. There are the reports. And particularly if you see in ancient people, okay, ancient Indian peoples, they are familiar with the silver nanoparticle and the copper oxide nanoparticle. Okay, somebody is, I think Sonaune sir is presenting. That is the problem. So I request Sonaune sir, stop the presentation. Sonaune sir. Okay, so if you've seen the silver nanoparticle and the copper nanoparticle, where is the recent use? If you've seen the recently they are used in the RO system. Okay, RO system that we are using the homes. Okay, so copper RO. Can't make the much more advertised. So if you send the copper metal, when it is oxidized, we get copper oxide nanolayer on it. And these copper nanoparticles are released from that, and they act as a antibacterial. They act as a antibacterial. So if you see in the Tata Swatch, okay, Tata Swatch, it consists of the silver nanoparticle, Ag nanoparticle, and they are embedded in the polymer. And there is a slow release of this silver nanoparticle in the Tata Swatch, and which causes the purification of water. Purification of water. So like this, you have the number of applications of silver nanoparticle. So in this hands-on training, I'm going to represent a very simple method for preparation of silver nanoparticle. Okay, there are many reported methods, biological methods are there. That is, the plant extract can be used, biological method means, and the chemical method. So here,
शिंदे सर आवाज कट होतोय शिंदे सर.
Hello.
It is clear.
Hello.
हाँ सर येतो आता बोला.
Okay. So what we require? We require concentrated ammonia solution. We require air-grade glucose, or you can use the apple juice, or you can use the plant extract material. Okay. We require 0.01 molar silver nitrate solution in distilled water. So very simple, just weigh 0.169 gram air-grade silver nitrate and dissolve in 100 ml. So here, what is the role? Concentrated ammonia, it reacts with silver and it forms AgNH3 twice complex. Okay, the complex is formed. If complex is not formed, the reduction do not take place. Another thing, we have Ag plus ion, we are converting to the metal, and this reaction take place in the alkaline medium. So what is the role of concentrated ammonia? Role of concentrated ammonia is to maintain alkaline condition, and purposefully we have taken the ammonia. I will explain after what is the use. There are many chemicals. You can use the vitamin C, you can use the glucose, you can use the formaldehyde or any aldehyde. Like this, there are number of biological material, and all they act as a reducing agent. All they act as a reducing agent. Then silver nitrate. Yes.
Sorry to interrupt you. We are not able to see the PPT. Please share your screen.
Yes, I have shared, but someone is sharing from their side. I think Sonawale sir is
you are
there.
explaining, but we are not able to see anything from the screen.
This thing, sir. Sir, we can see.
Pin up.
This sir, we can see.
Just pin up me.
It is visible.
Or Bange sir. Problem is there. I
Hello.
think so. Not. You have extension. Okay.
हॅलो शिंदे सर.
हा
सगळ्या पार्टिसिपंटला आता मराठीमध्ये सांगतो मी की जी स्क्रीन आहे तुम्ही पिन करायची आहे. जे प्रेझेंटेशन शिंदे सर करतायत, प्रेझेंट करतायत, त्याला पिन करा. कुठलाही रिसोर्स पर्सन असेल तर जेव्हा तो प्रेझेंट करतोय त्याची जी प्रेझेंटेशनची स्क्रीन आहे त्याला पिन करायचं, तरच तुम्हाला दिसणार आहे. अदरवाईज ती तुम्हाला दिसणार नाहीये. आणि दिलीप सोनवणे नेहमीच ते प्रेझेंट म्हणून
प्रेझेंटेशन केल्यामुळे
स्विच होतं. दिलीप सोनवणे जवळपास 2-3 वेळा तसं केल्यामुळे ते तुमच्याकडून बाजूला गेला असेल.
बरं ठीक आहे, मी परत शेअर करतो.
शेअरिंग इज ऑन युअर शेअरिंग ऑन.
शिंदे सर, युअर शेअरिंग इज ऑन. आम्हाला दिसत आहे ज्यांना
फक्त पिन
दिसत
करायचंय.
नसेल त्यांनी त्या विंडोवर जा आणि तिकडे पिनचा जो सिम्बॉल आहे त्याला क्लिक करा.
Okay, sir.
Yes.
Continue, sir. Continue.
Okay, I'm sharing again, so don't have problem.
Right, we are up to.
Okay, so we have explained this slide. So here particularly we have used the AR grade glucose. When glucose is oxidized, we get gluconic acid. Okay, it is not toxic one. When we are heating the solution, concentrated ammonia get totally evaporated, and whatever we get behind is the silver nanoparticle, which can be used for the biological activity. So when glucose is used as a TC agent, whatever remaining behind is the gluconic acid, which is totally non-toxic, and that is the purpose. So non-toxic one, so this method can be also called as a green chemistry method. Okay, so what we require? Transfer 40 ml distilled water in the 55 ml hard glass test tube. So I will take up the water, okay, in the pipette, and I will transfer water into the hard glass test tube. Okay, then we have a step 2. With the help of 1 ml pipette, you have to pipette out the required quantity of the silver nitrate solution, such as 0.2 ml, 1 ml, like this. So I have pipetted say 1 ml, and I am transferring silver nitrate solution into the hard glass test tube containing water. Okay, step 2. Now step 3, we have to add concentrated ammonia with the help of pipette, and we have to withdraw 2 ml of concentrated ammonia and transfer into the reaction mixture. Transfer into the reaction mixture. Okay. The next step, we require 0.1 gram glucose solution. By mistake here, I have put up the 1 gram. It is 0.1 gram. Okay. So in the copy paste, this is the problem. I have just pasted the word slide, but it should be 0.1 gram. Okay. So I will weigh the glucose. Okay. So by mistake, it is 1 gram, but it should be 0.1 gram. Okay. So we have to transfer the glucose solution into the reaction mixture. Okay. So just stir the reaction mixture so that we get the homogeneous mixture of the reagents. Okay. So we have to take a beaker and nearly take start the heating, put up the reaction mixture. Okay, and now see the color change. You have to observe for the color change what happens on heating. Yes, you can see the test to be slightly yellowish in color. That indicate that on heating the formation of nanoparticle has started. Yes, slowly yellow color will appear, and the intensity of yellow color will go on increasing with time and the heating. So very simple method, the green chemistry method for the preparation of the silver nanoparticles. Okay, so you are getting slowly intensity is going on increasing, and you will get golden yellow colored silver nanoparticle on heating. Okay, so we get the silver nanoparticle, and we have to confirm the band gap of the silver nanoparticle. So what you have to do? Ultimately record UV visible spectrum in solution mode. Okay, so yesterday you have seen this. Just transfer the blank transfer the sample. Okay, close the compartment and start recording the sample. Okay, start recording the spectrum.
Video off.
Okay, so you will get the such a beautiful spectrum for silver nanoparticle from the peak. Okay, here it is the peak. Here it is the peak, and it is in between 400 to 420 nanometer. Depending on particle size, even it can go up to the 450 nanometer. Depending on particle size, the lambda max in the visible region, this peak position in the visible region changes. And absorption at this wavelength from 400 to 450 nanometer is responsible for beautiful golden yellow color of silver nanoparticle. Okay. So in laboratory, we have prepared the silver nanoparticle, and you can see the beautiful photograph early in the morning. I have started my work and I have prepared this 5 different size nanoparticle. Okay. So the for this I have used 0.2 ml, 0.1 molar AgNO3, second one 0.6 molar, 0.6 ml, 0.01 molar AgNO3, it should be not 1. Third one 1 ml, 1.5 ml and 2.0 ml. All we have not 1 molar AgNO3. Okay, same procedure whatever I have explained, that procedure is used, and these silver nanoparticles are prepared. Okay, so just you can see the video. Okay, so I have taken these 4 test tubes. Okay, and I have placed them in a water, and burner is on, so heating is going on. In first step, I am adding 40 ml water in each of the test tube. 40 ml water in each of the test tube. Okay. Second step, I have to add a silver nitrate. So I am pipetting out a silver nitrate solution. So I have just shown the 4 test tube. The first one, 0 number, I have not shown here. So this is test tube 1, 0.4 ml or 0.6 ml. Okay. It is 0.4 ml. Then we have 1 ml silver nitrate solution I have added. Okay. Like this, in third test tube 1.5, in fourth test tube I have added 2 ml. Okay. After addition of silver nitrate, we have to add concentrated ammonia. So I have 2 ml pipette, and with the help of 2 ml pipette, in each of the test tube, I will add 2 ml concentrated ammonia. Okay. Concentrated ammonia. To make alkaline condition, particularly ammonia is used. We can use NaOH or KOH also, but it do not get evaporated. When we are heating, whatever the excess ammonia is there, it get evaporated. So like this, in each of the test tube, I have added 2 ml concentrated ammonia. and simultaneously heating is going on. Okay, since silver nanoparticles, they are formed above 70 degrees Celsius only. Below 70 degrees Celsius, they are formed, but they took a lot of time, such as 1 to 1.5 hour. The next step, I am adding 0.1 gram glucose in each of the test tube. 0.1 gram glucose in each of the test tube. Okay, then we have to stir. Okay, stir the content of the test tube. So like this, I have stirred the content of all the test tubes when heating is going on. Okay. So it takes some time. So I will take the video forward. I will take the video forward. Okay, so nearly in 20 minutes, nanoparticle formation has started. So I will take forward so that you can see the formation of silver nanoparticle. Okay, yes, you can see the test tube number 1, its color has changed. It becomes yellow in color. Yes, you can clearly see test tube number 1 is yellow in color. This type of sir. Yes. Okay, it is clearly visible. So slowly all the test tube will convert. If uniform heating is there for all test tube, then you will find that the 4 number will be converted yellow, then 3 number, then 2 number and 1 number. But 1 number test tube is heating fast. Okay, so you can observe the intensity of color also. It is changing with time. Okay, now 4 number is started converting to yellow. So nanoparticles are forming in the boiling condition. Now the 3 number. Now the 3 number. So you can see the intensity of the color. Depending on concentration, the intensity is changing. This one of the effect is the concentration effect and another is the size effect. Another is the size effect. 3 number is converting yellow. Okay, now you can see the 2 number also. Okay, and here you can see now the changes. Test tube number 1, less intensity. Test tube number 2, increase. Test tube number 3, till more intensity, and the 4th one is having the highest intensity. Okay, I will take forward. Okay, now for 0 number, I will take forward, and when color is formation is started, I will run the video from that point. Yes, you can see the yellow color is appearing. This is the lowest concentration, 0.2 molar, 0.01, 0.2 ml, 0.01 molar silver nitrate. Okay, so in the lower part here, you can see the colors of the 1, 2, 3 and 4 number solutions depending on concentration and the size. There is change in color. There is change in color. Okay, so very simple method. Okay, and the green chemistry method. Whatever the things are there, the silver nitrate is there, ammonia get evaporated, and you will get the gluconic acid, which is non-toxic. So for biological activity, such a nanoparticles can be used very easily. Okay, so this is the simple and the easy method for the synthesis of silver nanoparticle. And here you can see the changes in the color with respect to the concentration and with respect to the size of nanoparticle. Okay. Then you can find out the band gap. We have the UV visible spectrophotometer. Okay, the screen is not appearing properly when we are making the video, so I have directly recorded the UV visible spectrum. So you can see for the AG0, this is the peak in the visible region, and maxima is quite here. It is around 395 nanometer. 395 nanometer, and this is in the ultraviolet region. This is in the ultraviolet region. So AG0 we have. Then AG1. Okay, so whatever the peak which is not clearly visible at 0.2 ml, it is clearly visible, and you can see this is the absorption around 400 nanometer, and here again lambda max is around 395 nanometer. So 1 and 2, that is 0.2 ml and 0.6 ml, the absorption is at a same wavelength, so particle size is similar. Now the third one, that is AG2. You can see the key difference here. AG0 absorption at 400 nanometer is very less. AG1 it is increased. AG2 it is still increasing, and you can see it is shifted. Okay, lambda max is shifted, and it is around 406 nanometer. 406 nanometer is the lambda max for 1 ml silver nitrate solution. Okay, now the next one, see very carefully. The increasing size increases the peak intensity in visible region, and whatever the peak intensity in UV region, it is going on down. It is decreasing. This is the size effect. This is the size effect of silver nanoparticle. And if you see the lambda max here, it is 411 nanometer. It is 411 nanometer. Then the last one, you can see clearly, this is still decreased. Absorption in ultraviolet region, it is still decreased, and absorption in the visible region, it is increased. And here lambda max is 417 nanometer. So we have the lambda max 395, 406, 411, and 417 nanometer. So why this difference is there? Yesterday I told you the band gap. So if AG0, this is the band gap, then for AG1 it is similar. Then we have AG2, AG3, and AG4. So this band gap is decreasing. Delta E is decreasing, and as delta E decreases, the absorption takes place at longer and longer wavelength. Longer and longer wavelength. So you can prepare the silver nanoparticle. Say you take 4 ml, 0.01 molar AgNO3 and repeat similar procedure. Clearly you will get turbid solution. Turbid solution. Okay, and turbidity appears, all these solutions are clear. Turbidity appears as the particle size increases. But many times when you use higher concentration, you will get the formation of silver film on test tube. Okay, so whatever in chemistry we are taking the silver mirror test, it is due to the formation of silver nanoparticle film on inside of or on the inner side of the test tube. Okay, so this experiment, after trial and error, 2 to 3 days trial and error, the beautiful experiment is designed and we get the shift in lambda max for the silver nanoparticle. Okay, if anybody has doubt, you can ask now.
Okay, dear participant, if you have any doubts regarding the synthesis process of silver nanoparticles,
okay, you can ask doubt. Otherwise, I will go to the copper oxide.
Sir, what should be the concentration, sir, to start with?
Yes, I have taken point. So I will just show the calculation to you people. So you can use the formula. N1 V1 is equal to N2 V2. So N1 we have 0.01 molar. Volume we have taken 0.2 ml. The N2 we have to calculate, and V2 we have 40 ml plus 2 ml plus 0.2 ml. Okay, so 42.2. So 0.01 into 0.2 upon 42.2. So we can calculate this. Point not 1 into 0.2 divided by 42.2. Okay, so 4.7 into 10 raised to minus 5 molar. So low concentration is required. Okay, at this, even you can use 0.1, but yellow color will be very, very less. When we start with 0.2 ml, this will be the concentration. Just once again, I have to confirm. Yes, it is 10 raised to minus 5. Okay, such a very low concentration. So approximately it is 0.0005 molar silver nitrate, and you can get beautiful silver nanoparticles. Okay, which will absorb more in ultraviolet region, and when this concentration is nearly 10 times of this, okay, not 5 molar, then you will get the silver nanoparticle which has much more in visible region and less in ultraviolet region. Yes, anybody has doubt? So now one more point, you can calculate. You can calculate the lambda max, band gap. You can calculate the band gap. Band gap is equal to 1240 upon lambda. So 1240 upon 395 nanometer, you will get value in electron volt. 1240 upon 406, 1240 upon 411, and last one 1240 upon 417. So there is small change in band gap, and due to small change in band gap, you can see the difference in color. You can see the very clear difference in color of the silver nanoparticle. Okay. If anybody has doubt regarding the silver nanoparticle?
Sir, yesterday also we have seen the calculation of band gap.
Yeah.
And we have seen the example of silver only. Then it was in the form of suspension. So is there a possibility of scattering of light?
Okay. Yesterday we have seen the solid state. For example, ZNO, or you have the silver nanoparticle in the solid state. Then we are using the reflectance mode, and then we have to use the KM plot. We have this silver nanoparticle, and the solution is clearly transparent. You can see here. Solutions are clearly transparent, and hence we have used the solution mode. We have not used the solid state. So whatever the regular invisible spectrophotometer, we are recording for the absorbance of solution state, same is used, and absorbance spectrum is recorded over 250 nanometer to 600 nanometer. Okay, so solution state and the solid state, this is the difference. We don't require here KM plot. Yesterday we have seen the equation. Okay, delta E is equal to h nu upon lambda. So h nu is equal to 1240 in terms of the electron volt. In terms of electron volt. Yes.
Sir, at what wavelength we will say that we have got the nanoparticles exactly for silver?
Okay, so when you get the absorbance around 400 to 450 nanometer, you should get very intense peak. Then and then you can say that silver nanoparticles are formed. Many times when we use the biological material, whatever the enzymes are there, they get converted to the brown color or the yellow color. And when biological material is used, in this case, whatever the degradation product of enzymes are there, they interfere in the determination of the lambda max, and many times this yellow product of the enzyme degradation or the chlorophyll degradation that overlap with this, and many times with biological material, clear-cut absorption due to silver nanoparticle cannot be recorded. Okay.
Sir, one more question.
Yes.
Sir, we have a suspension here. So is there a possibility of a scattering of light? Because we are recording absorbance.
Yes, ultimately, if you see in analytical chemistry, silver nanoparticles are used for the analysis of many toxic, even silver, cadmium, some chlorinated pesticides they are determining, and they are using the scattering properties of silver nanoparticle. You can take the Raman spectrum. Very beautiful Raman spectrum we will get due to the scattering from the silver nanoparticles. And by using the Raman spectroscopy, you can analyze the many of the toxic substances. Even in the milk powder, if you taken the milk powder from China, okay, they add the toxic substance, okay, and it is melamine. Melamine. The 2 years back, this is the case. Whatever the milk powder that is imported from the China, that consisting of melamine, and the melamine is determined by using the silver nanoparticle, by using the Raman spectroscopy, and even ppm level of melamine can be determined by using the silver nanoparticle and by using the Raman spectroscopy, where the scattering effect is used. Yes.
Sir, is there any effect of scattering on band gap calculation?
Yeah, you can use the photofluorimetry. The fluorescence spectrum can be recorded of the silver nanoparticle.
Okay, thank you, sir.
Yes. So, If you don't have question, we'll go to the next part. That is the copper oxide nanoparticle. Again, very simple method I am presenting for the biological as well as for the chemistry peoples. Since many times when we hear the name nanoparticle, there is afraid how we can prepare the nanoparticle, how we can obtain the nanoparticle, does particle obtain are really nanoparticle. So I already told you that at the start of the lecture, these are the biologically active, and the as far as the mixed group is considered, I have taken this. So it is very simple method to prepare the copper oxide nanoparticle. What we require? We require 0.2 molar copper sulfate solution. Okay, and I have taken 400 ml. 0.2 molar copper sulfate solution, and I have placed it for boiling. Okay, I have heat this solution to the boiling. Then I have prepared 0.5 molar KOH, very high concentration is required. 0.4 to 0.5 molar you can easily obtain, and boil the KOH solution also. Heat the KOH solution also. Okay, and now the procedure is very simple. You have to dropwise add this hot KOH solution into the copper sulfate solution. This is the procedure. Dropwise we have to add. If temperature is less than 60 degrees Celsius, you will get blue colored precipitate. You will get blue colored precipitate. If temperature is 70 degrees Celsius, initially you will get brown color, which is converted to blue. Then 80 degree or 90 to 100 degrees Celsius, temperature dependent, you will get brown color, precipitate directly. You will get brown colored precipitate directly, and it will not converted to yellow one. So if you want direct nanoparticle, keep the solution above 100 degrees Celsius, that of copper sulfate, and add hot KOH solution. Don't add cold KOH solution. Okay, temperature also affect the particles. I will just show the small video that I have prepared in the morning. Okay, so I have this copper sulfate solution, and on this side KOH solution I have kept for heating. So I will just take the video forward. Heating is carried out till the temperature of copper sulfate solution is above 90 degrees Celsius. Okay, so it has taken nearly 10 minutes time. Yes. Okay, now at this point I have started addition of KOH solution. Here you can see the blue precipitate is formed, and when it goes down, it is converted to the brown one. Okay, so temperature is less as such. It is around 70 degrees Celsius. Okay, here drop is placed, and at the drop you will get blue precipitate, and it is slowly converting to the brown one. Okay, now temperature is increased, and you can see the formation of copper oxide nanoparticles. Blue is there, it is copper hydroxide. The blackish brown color is there, it is copper oxide. Go on adding KOH dropwise. And we require excess of KOH for complete conversion of copper sulfate to copper oxide. We require the okay, so I will just take forward. Okay, so when excess of KOH is added, you can see completely brownish or the blackish precipitate is there. Okay, so we have to go on heating at 70 degrees Celsius, since there may be the presence of some copper hydroxide also. So we have to heat nearly for 3 hours on boiling water bath, not directly. Once copper oxide is obtained, you have to put this flask in the water bath, and on boiling water bath, we have to heat for 3 hours or on low flame, heat for 3 hours. Okay, then filter it and dry in oven. Filter it, wash with distilled water. Okay, then wash with ethanol, or you can suspend it ethanol for overnight, then filter again and dry in oven at 60 to 70 degrees Celsius for 3 to 4 hours. You will get the copper oxide, which may be amorphous. Then finally you have to heat in muffle furnace at 190 to 200 degrees Celsius for 3 to 6 hours. You will get the copper oxide nanoparticle. Okay, so we have not taken the XRD, but I have taken the UV visible spectrum. Okay, so again I have taken the UV visible spectrum in the suspension state. If silver nano like that, just I have the name is incorrect one. It is only the copper oxide nanoparticle, not copper Ag. So you can see the lambda max is around here. So it is around 480 nanometer. Lambda max is 480 nanometer. So 1240 by 480, you will get around 2.5 electron volt. Okay, this is the band gap of typical copper oxide nanoparticle. This indicate 2.5 is the characteristic value for the copper oxide. So copper oxide nanoparticle, again, they can be evaluated for the biological activity or for the other purpose, such as degradation of the dyes. But for degradation of the dyes, the copper oxide nanoparticle, they are not much more suitable, since activity is very low. Like that of TiO2 and ZNO, the activity of copper oxide is very low. For ZNO, TiO2, photocatalytic activity is high. This copper oxide nanoparticle can be used for the adsorption of toxic metal, then adsorption of polar organic pollutants such as the pesticide. And you just put the word in the Google search, what are the applications of the copper oxide nanoparticle? Much more information you will get. Okay, rather whatever I am saying, I am just giving the few ideas to you, but you can get the number of ideas. Okay, now what you can do, okay, in the research, you can prepare the copper oxide nanoparticle, silver nanoparticle, evaluate biological activity separately of copper oxide, such as for the antibacterial, silver nanoparticle separately, copper oxide, and then add silver nanoparticle in the copper oxide. So silver nanoparticle will be adsorbed on the copper oxide, and you can evaluate the activity. Okay, such a simple ideas you can generate when you have the silver nanoparticle or the copper oxide nanoparticle with you. Okay, with this I've finished my presentation. If you have a question, you can ask.
Okay, dear participant, please any doubts or any query regarding this presentation, please ask. Why Singh sir give the presentation regarding the preparation of this nanoparticle of copper oxide and silver nanoparticles? Because of this, the participants of the FDPs are mixed mob. That is a researcher from the life sciences and chemical sciences are there. And we discuss about the different technique useful for this characterizations of the different type of the component. Now, before that, we also very important things regarding this preparation of the nanoparticles, because of it is a interdisciplinary approach. Research go hand on hand. There is a cooperation between the biological sciences people and chemical people. Therefore, we generally prepare the different type of compound or nanoparticles in our laboratory, and we try to find out the different applications of this nanoparticle. Now, when we carry out the literature review, we observe that this is the application of the copper oxide nanoparticles are mainly utilized as the antimicrobial agent. Therefore, if anybody interested about the studies of the antimicrobial activities of these copper oxide nanoparticles, we are the life sciences people are better know the antimicrobial activity as compared to our chemical people. We are only expert in the synthesis of the nanoparticle in maybe colloidal or in the powder form. Okay, first we discuss about the synthesis of nanoparticle, silver nanoparticle in a colloidal form. And the second one, we discuss about the experiment preparation of the copper oxide particle in a powder form. Therefore, the powder form copper oxide generally used as the antimicrobial. They are used in hospitals due to their antimicrobial ability to kill more than 99.9% the gram positive bacteria as well as gram negative bacteria within 2 hours. This is the best applications of the copper oxide nanoparticles. Therefore, they are very important specific the interaction with the gram positive as well as the gram negative bacteria, and with a short time, not more time is needed for this interaction. For that, only within 2 hours, when exposed to this bacteria with the copper oxide nanoparticles, they kill. Okay, therefore, this is the suitable dose. We can apply this copper oxide nanoparticle to control or kill this type of the bacteria are there. Now, when we discuss about the silver nanoparticles, with the colloidal particles, they are the tremendous applications. They are these, for example, they include medical, food, healthcare, consumer, industrial purposes. Therefore, due to their unique chemical and biological importance, these are having a different optical, electrical, thermal, and biological properties of these nanoparticles. That's why this is the motto behind, or that is our idea behind about to show they're simple. These are the simple method. There are different complicated methods are available for the synthesis of the nanoparticle. Sindhu sir tried to develop or try to explain the simple chemical as well as the biological method to preparation of the silver nanoparticles and the copper oxide nanoparticles, which have the importance in the very importance in the biological field. Okay, that is the antibody. That's why the we considering the interdisciplinary approach, we try to synthesis about that. Now, okay, there is some beakers are also available with synthesis this silver nanoparticle, but the visualization this sir. Yes, sir. Okay, so these are the flasks. Can you see these flasks?
No, can't see.
Can't see, na? Okay, that's why we make photographs.
Yes, now you can see pin up me.
Pin up me. In this photo.
Pin up to me, you can see. Okay.
Can you see?
Yes, we can see flask. Yes, sir.
Oh, yes, yes. Now this is higher concentration. I now see the lower concentration colloidal nanoparticles. Okay, color changes. Can you observe the change?
Yes, sir.
Okay, now we see carefully. We observe the color change.
Yes,
okay,
sir. Yes.
The first one and last one. Now in between that, another 4 flasks are here. Therefore, we try to show the what type of nanoparticles are that, and it is color. How the color changes are there. It is depend again the particle size and concentrations of the reactant. Already Sindhu sir explained on how to calculate the band gap. Okay, I hope if any doubts or any query regarding this presentation, any simple methods we want, biological method we want to discuss with the Sindhu sir, if ask the question. Anybody?
Sir, simple methods for detection of nanoparticles are apart from visual observation.
Yeah, ultimately, at least you have to do the UV visible spectroscopy. Okay, the color is there and the UV visible spectroscopy is there. So otherwise you have to go to the XRD and the SAME and TAPE. Okay, with the help of SAME, TAPE and XRD, you can accurately determine the size and you can accurately predict the size in the nanometers.
Okay, sir.
Hello.
Hello.
Hello, sir.
Yeah.
Sir, my question is very basic. What is the basic difference between properties of normal silver and nanoparticle of silver?
Yeah, if you see the normal silver, it is shiny, that is a metallic color. We call it as a metallic luster. But if you see here, okay, if you see here, I will show you the camera. This is the reddish in color.
Okay, sir.
Okay, so silver metal, we have the metallic luster, whatever we are calling as a silver color. These are the silver particles, like that of silver metal, but color is different. It is absorbing in the visible range.
Visible range.
But when the silver metal is there, it is absorbed in the all regions, all wavelength. Okay, 200 to 800 nanometer, and hence it appears white in color. But it has a specific absorbance around 420 nanometer, which is different from the bulk nanoparticles. From the bulk size or the large size.
Okay,
Thank
good
you,
question. Regarding that, what is comparison between the simple silver nanoparticles and simple silver particles and nano silver particles of the silver? Okay, sir, please repeat the question, sir. Mangesh, sir.
How stable are these nanoparticles?
Yeah, if you see the silver nanoparticle, at least they will remain in the suspended form for 3 to 4 days. Since it consists of ammonia, and ammonia acts as the capsulating agent over the silver nanoparticle. If you evaporate the ammonia, slowly they will settle. Within 3 to 4 days, they completely settle, and you will get the golden brownish color, golden to brownish color to the silver nanoparticles. Solid state. Otherwise, you can centrifuge at 10,000 G. Very high speed centrifuge is required, around 10,000 RPM per minute. Then and this silver nanoparticle can be isolated. Otherwise, they cannot be isolated.
Okay, thank you. Okay, I hope your doubt may be clear. Okay, any other? Now there is a time is running fast. So I appreciate the participation of this all delegates. Or there is an interactive session of the demonstration of this silver nanoparticles and colloidal nanoparticles and copper oxide nanoparticles. Therefore, I thanks to all participants to interact and asking some queries or doubts. And so also thanks to Shinde sir to resolve the whatever queries they are raised by the participant. Now I request one of the participant, Dr. Professor Kalnande from Puttur College to give the vote of thanks.
Am I audible?
Yes.
First of all, I am very thankful to the organizer for providing me this opportunity to propose the vote of thanks to Dr. Shinde sir. It gives me a great pleasure to propose a vote of thanks to Dr. Shinde on behalf of Professor Ramkrishna Model College Akurdi and HRDC Pune, and on behalf of all the participants of this faculty development program for conducting informative technical session. Dr. Shinde sir gives us the information about green synthesis of silver nanoparticle and how to determine their band gap. They told us about the method using the silver, ammonia and the glucose for the synthesis of silver nanoparticle, and how the color of nanoparticle, it is depend upon the size effect and the concentration effect. Also, they explain the color dependence of band gap. They explain that size of the nanoparticle increases, then band gap, UV band intensity decreases and visible band intensity increases. They also explain the synthesis of copper nanoparticle or copper oxide nanoparticle by using the copper sulfate and the potassium hydroxide. I also thankful to all the participants who raised the question, and Dr. Shinde sir gives the answer to all that questions satisfactorily. Therefore, I am thankful to all the Shinde sir again and all the participants. Thank you very much.
Okay, thank you, Yogesh, for the giving a vote of thanks or appreciation of efforts taken by Dr. D.R. Shinde. Last 3 to 4 days, day before, he tried to develop a new methodology to synthesis of the silver nanoparticles, and he finally succeed, and we demonstrate today in front of you. Okay, now I request Rasai sir, any suggestions or any Rasai sir?
एक मिनट
Any announcement regarding the tomorrow's schedule? I request the FDP coordinator, Dr. Suresh Rasale, give this some instructions or any change schedule from the tomorrow's.
So, any one of you?
Okay.
Tomorrow's schedule.
What Rasale?
Tomorrow's schedule start from 9:30 a.m. sharp. So join before 9:30. And tomorrow's schedule is given on your WhatsApp group. See the schedule. Also, fill up today's feedback form and also submit your assignment, second assignment, which is given on your classroom. Thank you. We'll stop here.
Okay. Tomorrow. Okay, tomorrow sharp 9:30 join the session.
Yes, sir.
Yes, sir.
Bolo, Mahesh, bolo.
Rasal sir, do we get receipt of the payment that we have made for this FDP?
Yeah, we will send out last day. Your certificate as well as receipt, you will get it.
Yeah, okay.
Yes, after completion of FDP, we hand over these soft copies of the receipt as well as the certificate. And then we send also the hard copy of certificate by the speed post.
Yeah.
Okay?
Yes, okay.
Okay, have a nice day. We will meet tomorrow at sharp 9:30. Okay? Okay, thank you. Now,
Okay, thank you, sir.
Mangesh sir, stop it.
Thank you, sir.
Thank you, sir. Thank you. Thank you for nice session.
Thank you.
Thank you, sir. Thank you.
Thank you, sir. Thank you.